Hello?
Oh, yeah. Oh, yeah. All the chatter, please.
All right, so we're going to go ahead and get things started. I'm James Peterson. I'm Senior Director of Operations and Outreach for the Center for Physical Genomics and Engineering. And welcome to the Symposium on Physical Genomics. We are going to kick off the symposium with an introduction by our Associate Dean of Research Administration and Oversight. Yes, that's right. Dr. Eric Perot. He's been with the McCormick School of Engineering and the Department of Biomedical Engineering for a long time. I've known him since he was Director of Graduate Studies. So, Eric, please.
Thanks, James. Welcome, everyone. It's a pleasure to be here. I'd like to welcome you to this symposium today, especially, and thank those of you who have come, especially those of you who have made trips from various parts of the world. Before starting on the symposium, which I think is really exciting, I'd just like to give you a little bit of a background on the Center for Physical Genomics and Engineering. The center started in 2018, and in the McCormick School of Engineering, we were thrilled to launch this center because it was an opportunity to take what had been one of our most productive research laboratories, Vadim's laboratory, and really help them expand their footprint. And the center has been tremendous in many different ways. If you look at all the standard metrics, such as research expenditures and so on, they're consistently one of the top 2 centers in the School of McCormick Engineering. But I think much more importantly than that is the spirit of collaboration, creativity, and mentorship that they've instilled and brought and really grown from what was a productive laboratory to really a real gem or one of the crown jewels in terms of centers at Northwestern. The Center for Physical Genomics and Engineering has recruited exceptional faculty from not only engineering, but also the School of Medicine, the School of Arts and Sciences. They brought in collaborators to Northwestern from the Chicago area and far beyond. And this has created just a really vibrant community to study the cell, chromatin structure, nuclear structure, genetics in a way that hadn't been done on campus before. We've obviously had expertise in those areas, but what the center has done is really brought in a lot of engineering to that. And so I'd really, congratulate Vadim and his team on what they've done. And I look forward to what comes in the next 6 years of the center. Today, we have a really exciting program that's brought together leaders in the field. I'm really looking forward to that. And I'm looking forward for allowing you to see some of the work that the center has done in the poster session at the end of the day. So I think you're in a treat today. Welcome in for a treat today. Welcome again, and I'm looking forward to a great program.
All right, I'm Vadim Bachman, and I would like to welcome you once again to this, what promises to be a fantastic event. I know I'm going to enjoy it greatly, and I hope that all of you will do too. The most important thing of what I think so special about this event is the people in the room, the audience, the guests, and most importantly, the speakers. As Eric said, we have really fantastic, incredible lineup today. I'm going to cherish every minute of your presentations. I hope everybody will enjoy it too. The idea here today is to do something that is not typically done in regular symposia, which tend to focus on one particular area. We want to bring in people from different walks of scientific lives, really, to have more diversity of opinion. And the talks you're going to hear today will range from what can be described more of a fundamental biology to engineering, to biotechnology, to imaging, et cetera, et cetera, et cetera. And I think that's one of the exciting aspects that I find so interesting in the field of physical genomics, that it is an example of a convergent science, which makes certainly more difficult to pursue, but I think so much more rewarding. And I think every speaker, every one of the speakers today has done and is pursuing transformative convergence research. true disruptors, every one of them. Here at the Center for Physical Genomics, we are trying to accomplish something along those lines, bringing biologists and engineers and physicists and physicians together. We are lucky to be funded by a U54 grant from NIH. James, whom you had pleasure seeing make an introduction, sitting right there, is directing the first training program for graduate students at the interface of physics and genomics, funded by NIH. And some of the students who are trainees are here, sitting in these chairs, and we'll have poster presentations later today. Let me say a couple of words about the logistics, what's going to happen today. We will have talks, and we'll get to the talks as soon as possible, because you're not here to hear me, right? You're here to hear our wonderful presenters. So we'll get there in a second. There will be no Q&A during the talk or right after the talk. However, we have a dedicated time right before lunch, after the first 3 talks are done. There's a table right here. We have a panel. Our speakers will be here at the panel, and you will be able to ask any questions. And I mean really any question. It doesn't have to be a question specifically related to anybody's talk. It can be, if you want to ask a question about world peace, please do so. Right? That's the whole idea. We have incredible people with the incredible breadth of knowledge and ideas, and so it's free for all. Ask any question you want. That's that. And I will try to moderate this to the best of my abilities. We'll have lunch brought in right across the street, right there, in a new building that is called Simpson Query Institute, dedicated to biomedical research and biomedical engineering in particular. We have a poster session, quite a few exciting posters brought in by students who work at Northwestern, McCormick, CPG Center, but also we have several posters from faculty researchers who are not part of Northwestern at all. So please feel free, grab some lunch, enjoy the posters, then we'll be back. We have 2 more talks in the afternoon. We have another panel, and then we'll have a reception in the end of the day. So without further ado, I would like us to get to the main part of the program. And what a better way to start the program than with our keynote speaker, Clodagh O'Shea. Clodagh has had a remarkable career. She's a Wicklow chair and professor of molecular and cell biology at the Salk Institute, a professor at the UCSD, where she directs the reimagining medicine biology. Redesigning it. Sorry, I knew it starts with an R.
I prefer imagine.
Okay. So let's call it Reimagining Medicine and Biology initiative, which stands at the forefront of innovation in biomolecular sciences. Clodagh received her PhD in immunology from the Crick Institute and Imperial College London. And in a way, I would say, well, correct me if I'm going to say something wrong, but I feel like the understanding of viruses has been a kind of a recurrent theme in what you do. Is it fair to say? Right. Designing viruses, using viruses as drones, for the lack of a better word, as ways to understand tumor targets, how tumors work, how other diseases work, ways to understand genomic circuits and how to fix them. So very innovative use of viruses as both therapeutic and diagnostic agents. But Clodagh's research has progressed and evolved well beyond what we can call just immunology, and includes state-of-the-art computational genomics, understanding the circuits that drive, in particular, cancer cells that get dysregulated, ways to fix them, imaging technologies, in particular. A few years ago, Clodagh's lab pioneered chromatin electron microscopy tomography, a groundbreaking technique that is poised to address one of the long-standing, or help address one of the long-standing problems in biology, very fundamental problem. How exactly does the 2-meter DNA fit inside a fairly small 10-micron nucleus, and what the significance of this organization actually is? This is one of the problems that really converges, that brings together imaging and physics and engineering and biology and computational science all together. And I think Clodagh's research epitomizes what convergence research is and what it can do. Clodagh received so many awards that if I were to try to list all of them, I think I would probably take us all the way to lunch. So I'm not going to do this, but I mean, just mention a few. She's a recipient of awards from the American Cancer Society and National Academy of Sciences. Many others. There's a very long list. There's a very long list. Anyone in particular your favorite?
They're good.
They're all good. All right. Very well. Long list of awards, very well deserved, of course. So without further ado, I would like to introduce our renowned keynote speaker, Clodagh Shea. Thank you so much. It's a pleasure to have you.
Thank you so much for doing. Thank you. Your renowned freelance screws this up. I see it. The PowerPoint. Okay. I just need to do. All right. Oh, perfect. Excellent. Thank you. Okay, so first of all, thank you so much. It's a real pleasure to be here. I'm a huge admirer of Vadim's work, and we've for many years, and actually what you guys have been doing here, and so it's just fantastic to be here and to be able to discuss all of this in person. And so yeah, so what I'm going to do is I guess this will work. I guess for you guys. To discuss mostly unpublished work and to throw out some ideas, but also sort of that I think reflects sort of the amazing kind of initiative and interfaces that you guys are at here, basically in terms of physics, engineering, chromatin biology, and also cancer biology, which is really very much at my heart. So kind of as Vadim said, as I said, for the graduate students, a lot of people will call me an interdisciplinary scientist these days, but mostly my graduate and postdoc advisors just sort of suggested that I lack focus. But now I'm interdisciplinary, but I just lack focus. But the overall goal of my research is really to identify critical growth regulatory targets and to try and really use this knowledge to impact and improve patients' lives, right? And that would be all of our goals. And viruses are just really sort of, how does a small genome take over a cell, right? And they're very clever. I'm not as clever, so sort of follow the virus. And they actually impinge on sort of the critical circuits that drive growth deregulations, the same processes, and they converge basically on these circuits. And that led to really the identification of many of the key targets and sort of continue to this day. So in terms of sort of understanding these targets, well, structure determines function. I was originally trained as a biochemist. And one of the key kind of sorts of challenges is really trying to understand structure in situ. And that required basically the development of sort of new technologies. And I'm going to actually speak mostly to that today. And then really kind of, I think the key challenge for us all is really, can we sort of leverage this knowledge, right, of these critical targets and structures to actually sort of realize the promise of genomic medicine, to redesign basically cells themselves and even viruses that can selectively seek and destroy them, okay? And that's actually sort of where my story began is it was actually, I was actually going to work with Jim Allison basically on the checkpoint inhibitors. This is around 2000, but I felt, well, that would be done. But could we actually find a virus that replicates in a tumor? And wouldn't that be sort of the perfect target to combine with immunotherapy? That's what led me to Frank McCormick's lab, who was where the first oncolytic virus was being tested in clinical trials to target the loss of the tumor suppressor P53. I thought it was a brilliant idea, I still do, but actually we didn't understand enough. And the reason it didn't work was there was another protein, which I then discovered in my lab in Salk, where I went to learn virology, which actually inactivated P53. So we never had that virus. And then this virus actually is what led me to the genome, and that it actually silenced DNA in a way that just couldn't be explained by the textbooks. And that led us to questions then about, well, actually, what is sort of the structure of DNA in the nucleus? And if we can understand that, again, can we begin to understand and realize the promise of genetic medicine? So it's fantastic to be returning to sort of the more fundamentals today, because hopefully we'll be in patients actually, which has taken me kind of away from some of this for the last 2 years, with first in human trials with some of the viruses that we've actually developed sort of in a company we span out basically for that. So now I'm back to the basic biology, and that's what I'm going to talk about today. So the DNA double helix, right, sort of provided basically the structural basis for how our genetic information is both copied and stored. But it's the structure of DNA in the nucleus that determines if it can be accessed and used, okay? But you can't crystallize a gene, right? And so that's the problem, right? How do we actually understand the structure of DNA in the nucleus, where you have basically sort of meter of DNA, more and more of DNA basically packed into a 10 micrometer nucleus. And really understanding this structure is fundamental to understanding basically the function of genes and the genome. And so, when I sort of was looking at the textbook model, beautiful work, by Olin and Olin's sort of with in vitro reconstituted DNA. Thank you so much. Sort of led to the idea that there was it was wrapped around nucleosomes basically, and then again based on in vitro modeling. Advance it again. Not sure if something has changed. I guess I can control it from here, maybe. Okay, back. Okay. Let's sort of the classic textbook model that sort of formed these hierarchical structures. But there's a reason it's a cartoon model, because none of these structures had been visualized in situ. In the meantime, sort of genomic sort of biology proceeded apace. And we know that basically DNA can be modified, wrapped around many different histone variants in nucleosomes. These are modified, and these can form sort of into these three-dimensional domains, TADs, LADs, basically nuclear bodies, all the way up to this. And the issue is that this spans multiple scales. And each sort of technique can sort of interrogate one of them, right? Again, it was sort of both Feynman and Francis Crick said various versions of, if you want to sort of understand how something works, just look at it, right? But easier said than done, because again, we can sort of look at these as sort of individual scales, but what we really need to understand is how they're integrated inside the nucleus to give rise to function, right? With quantum computing, maybe we can take all these different dimensions and begin to understand it, but the cell does that actually by integrating structure in the nucleus. So what is the structure of DNA in the nucleus across these scales? And if we can understand that, can we begin to understand the basis of sort of cell fate and actually disease? And that's what really led us to develop sort of Chromatin, which I think sort of together with these other techniques helps fill a gap. And sort of it was actually a fairly simple idea, which was really, is there a DAPI for DNA? Because the problem was contrast, right? How do you create selective contrast of DNA in the nucleus to make it electron dense so that we can sort of visualize its structure, right? This is a silly video, my favorite movies. This is Indiana Jones. And this is sort of the Last Crusade. And kind of going over this bridge, which seems to disappear into the background. And he has this trick. And if you understand this, you'll understand everything. Okay. So he basically throws dust over the surface, right? And so that it now can become visible to everyone else. And that's basically what we did. Okay. That's fundamentally it, right? So how do we do that? To create basically a surface map of DNA and chromatin in the nucleus. So certain inorganic dyes, and actually just in molecules in general, have a really interesting property, which is that when you excite them, in addition to emitting a photon to go back to the ground state, they can actually undergo something that's called intersystem crossing and donate an electron to a higher basically orbit. And in the cell, that can actually be sort of donated to triplet oxygen to create highly reactive local singlet oxygen, right? So it produces basically this reactive oxygen right around it. And so, which can actually then be used to catalyze the deposition of that Indiana Jones trick across basically anything that's intimately associated with DNA within a few nanometers. So we screened for dyes and we found one, DRAC5, a membrane permeable DNA binding dye that binds to DNA. And what happens is you'll see us switching on the light here, basically. And in the presence of this other small molecule, DAB, that's the dust, it actually catalyzes it basically into a polymer that sort of sprinkles down on DNA and associated proteins. Let me be kind of, again, very clear what this is and what it's not, right? It's using DNA, but it's creating a surface map of things that are intimately associated with it, which can be nucleosomes and other closely interacting proteins, right? And so that's what's kind of interesting about it. You're not going to see helices or anything like that, but you will actually begin to understand sort of the general densities. And this polymer basically then, DAB polymer, is osmiophilic basically. So it binds to osmium, a heavy metal, which can deflect electrons and allow us to visualize it in the electron microscope. And this is all together with Mark Ellison's lab. So osmium basically binds to alkene groups. And so this is a classic sort of a TEM, a thin section. And you can see the membrane stained, and here's the nuclear membrane. And that was a cell kind of half inner field, just so you understand what you're going to look at in the rest of the talk. And you can see suddenly that the DNA sort of is black in here. But the problem with a thin section is that it's 17 nanometers, so you're projecting through this massive volume, and yet things we want to see are 2 nanometers and 10 nanometers, right? And that's again now where multi-tilt tomography comes in, right? And so basically, and obviously there are probably way better algorithms as well for doing this, but this is, we initially used sort of 8-tilt, multi-tilt tomography, which basically sort of effectively creates computational slices, which are about 1 to 2 nanometers. And then you can actually travel through the whole volume. And sort of the classic picture, this is of the nucleus, not dissimilar to anything you might have seen in the textbooks. When we take 80 slices, this was a joyful day, sort of resolved actually into this. You can see the gap in the membrane here because nothing sort of is stained. And these little black pieces in here are actually nucleosomes, basically, so they're consistent with the volume of the nucleosome, and an individual sort of free strands of DNA, right? But then joy turned to horror because, you know, as I said, I really was coming from a very different area. We really couldn't see these sort of higher order fibers, 13 nanometer, 120 nanometer fibers. Everything sort of fluctuated in a way I would call it a heteromorphic polymer between about 5 to 24 nanometers, basically. And so at that point, then we thought, well, let's look at mitotic chromosomes. This is obviously where it's most condensed, you know, sort of the sort of iconic image of the human genome, and yet actually also a black box, 50% protein, 50% DNA. But could we now peer into the depths if this stain sort of had sufficient resolution? And so, and I'm moving quickly through this because this is published, but just to give you a sense of where we're kind of coming from this. And what you can see was very beautiful. It's more densely packed together. But again, the sort of primary polymer structure per se did not necessarily change, right? So again, it was roughly between 5 to 24 nanometers. And I think that's kind of interesting, right? Because effectively, you know, if structure determines function, if there are only a few structures, then how can we actually achieve many different functions and all the different transcriptional outputs? I mean, look around this room, every single one of us is different and has a different phenotype. And yet, basically, the cell cycle occurs in the same way, right? Development has occurred in the same way. And that's remarkable, right? And I think that's so exciting, right? And basically, this comes again to the idea of sort of critical phenomenon and how things can come together actually to dictate sort of key processes, but also give rise to variety, basically. And disorder can give rise actually to order as well as chaos. So when you have a flexible and disordered chromatin chain, then it allows actually for many different structures. which can explain potentially the different outputs that one could have, and many different histone variants and things like that. It also explains potentially, if the primary polymer structure doesn't change as you go through mitosis, how that information can be inherited from one part to the other. Because if you change structure, what is remembered basically? And so these can be inherited because fundamentally it doesn't change. But now the rest of this is actually unpublished. So now the question is, well, that's all very well, but that 30 nanometer fiber and these 120 nanometer fibers were kind of comforting because they kind of provided an actual model for how RNA polymerase might be excluded from basically the DNA to be able to transcribe the genes. But if everything is the same sort of diameter, I mean, now what, right? How do we basically explain that? So we think about sort of DNA. Polymer is actually kind of a misnomer in ways because it's actually not really made up of a repeating unit. Sequence is actually kind of different in a way. Each nucleosome can be different, each of these kind of compositions. So in that sense, it's heteromorphic, okay? Second, right, you know, there's different models basically where H1 sort of tail interactions can mediate interactions between chains, which I think are very elegant. Protein nucleosome bridges, and you'll hear a lot as well about liquid-liquid sort of phase transition. And the thing is it one or the other, or how do these all act together? Is there sort of a fundamental underlying principle, right, that has to be simple, that can actually sort of explain that? So biology is always sort of, I think, best understood in the simple language of both chemistry and physics, with fundamental laws it must actually obey. And so I'll come from, again, a biochemistry sort of background, but it's been it echoes very much actually some of the ideas that Vadim and others have put out there as well. So there's a really interesting phenomenon that basically chemical and polymer scientists have been using for years in the plastics industry, which is that certain flexible, linear flexible polymers basically undergo what is called liquid gel. And I'm not talking about liquid-liquid, I'm talking about liquid gel state transitions at a critical concentration, which in a good solute is at about 20 to 30% vol-vol. And so effectively, as they approach the critical overlap or entanglement concentration, effectively they become entangled. So imagine you're in a tangle line and you can't actually sort of get away from each other, right? You all start to get enmeshed basically, and you kind of begin to move as a mass basically in a crowd, right? And so you begin to undergo sort of these, so you can merge basically from liquid to gel, and these can become actually volume excluding and have really interesting properties. So what in some ways, if it's simple, right? Because you know, if we all have different DNA sequences and different polymorphisms, and basically we can trace trajectories back to yeast and all these different organisms, what's the fundamental principle? How do we build on a fundamental principle other protein apparatuses that could actually sort of give rise actually to the fundamental phenomenon, but have different kinetics and regulatory mechanisms? And so one question then is, maybe these different mechanisms I just described in the previous slide, what if they actually just mediate sort of chromatin concentration, basically? They can drive chromatin concentration in different ways so that it can approach a critical volume, which would exclude basically the transcriptional apparatus, okay? So how do we do that? Well, the nuclear membrane and basically the lamina-associated domains are known to be silent heterochromatin domains. So why don't we just take a look basically at them then, because we can do that. And this is the power of the tomography. So you're going from one side of the nuclear pore now to the other. You can see the chromatin sort of associated with the nuclear membrane, see the loop as it goes around here. But it's kind of only when we started to render this that, you know, begin to understand it. And this is sort of what you would see if you were a transcription factor kind of coming into the nucleus or a virus. Sort of reminded me of kind of a kelp forest, actually, you know, sort of, and this is again, this A-B compartments where basically if 40% of the genome is there, begins to channel you sort of in towards the center, right? And sort of a discontinuous gel filtration column, what I might call it. And so what is the concentration then basically at the nuclear membrane? And so what we did, again, these are just surface rendered maps of the chromatin through slices. And you can kind of put basically an overlapping sphere because I'm a climber, basically. And contour maps make sense to me. And so basically it was like, can we create topographic, 3D topographic maps of the nucleus of the different volume concentration densities? So this is actually where we know there's this heterochromatin up here. And what's interesting is they really have very different concentrations. So this is around 40%. And then in towards the center, you're getting sort of in around here. So it's about, this is a somatic primary cell. It goes from about 20 to 45%. And here's mitosis. And you know that the chromosome at mitosis must move as a gel. That's the only way it can resist drag across basically the nuclear plate, okay? And so effectively, this is a really high concentration. What was fascinating to us is it's exactly sort of the same concentration, right? About 47% as that at the nuclear lamina, okay? Where again, that's the concentration where it can undergo a state transition and where it's transcription inactive. So what if it's that? And so that's sort of one of the, so we kind of come up, this kind of this idea of a chromatin volume concentration, and I'll keep it sort of at that. There are other parameters as well, but we could do with a lot of help modeling this. We are not physicists. But effectively, you can render this in 3D. And sort of, size heterochromatin can exist up here, and you kind of have this kind of continuum. And then basically mitotic chromosomes are here. And so what we've been doing basically is sort of then looking at basically stem cells at different states of differentiation to create maps to understand sort of what those look like. Constitutive heterochromatin, such as effectively chromocenters, polycomb bodies and things like that, epigenetic drugs. But today, given your engineering focus, I'm going to talk actually about an individual piece of DNA that goes from the silent to the active state. But the principles kind of come from that. So the issues with what I've shown you is that only a few percent, basically about 4% of the genome is coding. So what am I looking at really, right? That's the problem, right? What can I say it's active or not? So I really wanted to see a gene. I've always wanted to see a gene, okay? And with light, we can begin to sort of, this is Bo Wang's paper, Cas9, we can sort of look at its localization and position. And we could, sort of use basically a correlated light and electron microscopy to zoom in, but you kind of have a fine, where's Waldo problem, where it's like, okay, but where's the gene, right? And so it sort of struck us that what we really needed was effectively kind of a two-color EM. I needed a genetically encoded fluorescent metal nanoparticle. Because we discovered that actually once you permeabilize the cells, which explains sort of some of the 13 nanometer and other structures, you really sort of disrupt structure. And so the thing that you see in the electron microscope is densities and metals. So how do I create light with metal and density? And the answer to that is sort of to look to nature. And that we actually store metals in all of our cells across all kingdoms of life in a protein cage, which is made up by ferritin, which is a monomer. Well, this thing kind of does is it basically assembles over 24-mer into this beautiful octahedron cage, which reminded me of a virus. It permeates my thinking. And acts as a cage which can store 4,000 iron atoms in the center, which is naturally electron dense and can be seen in cryo. So I thought, well, what if we could take this and actually now sort of reimagine it, actually, and re-engineer it so that it actually can be a genetically encoded probe that would store iron in the center so I can see it, like a genetically encoded nanogold. But now I want to put things on the surface so it can find genes, and so it is fluorescent, okay? And that was very hard. And we nearly gave up. This was Gavin, because there have been several papers that have been published on this. And I think what they're seeing is quasi-equivalency, where they see steric hindrance because we were not successful in seeing them in the electron microscope. And the reason was this is a native gel when we were 3 years in and almost about to give up, where we took E. coli ferritin, and this is a native gel. It should run up here if it's a cage, but it doesn't. It doesn't assemble in mammalian cells, and then it doesn't store iron. And so then we just looked across kingdoms of life and horses for courses. For some reason, that actually assembles beautifully in the nucleus of the mammalian cell. And then it can store iron. So we're like, yes. But now we needed to synthetically engineer it and create something new. So we took green fluorescent proteins and nanobodies from llamas and things like that, put them all together. Mostly we got a mess until about 300 goes in. And this was really, again, somewhat structure-based design and a little bit of directed evolution to get something that would assemble a proper cage at the top of a gel. So what the idea is then, we call this fire nano for fluorescent iron EM nanoparticle. And you can kind of put a nanobody or anything you want here, actually, really. A fluorescent protein, and then basically this is your iron cage. So you can genetically express that in cells, and it can go to the nucleus if you want with an NLS. And the idea was like, if you have a Cas9 targeted gene or locus, it would actually then find it, right? And actually give you a mark, that specific DNA sequence, okay? So before, with any unknown technology, you should test it against a known. And so to do that, we looked to this beautiful system developed by David Spector, which has been fundamental, and Rob Singer and others, to really sort of understanding the dynamics of RNA export and transcription and things like that. And what it is that there's 200 copies. I want to be very clear. There's 200 copies of a transgene which are integrated into a single locus, 1p36. And these have basically lac repeats, which can be bound by lac GFP. So that allows you to see a spot. And there you go. But then basically, so we basically made a fire nano, which could bind to GFP, but was red. So there you go. We're very happy with that. Forms a nice spot. And what's really cool about this locus is that it's actually sort of facultative heterochromatin. But in the presence of doxycycline, it has a teton promoter. It turns on a CFP peroxisome protein and an RNA, which has MS2 transcription, which become important. So we wanted to make sure we weren't perturbing transcription with our new device that we had engineered. And so what you'll see us now doing is add doxycycline, and the fire nano and lac GFP is there. Kind of expands a little bit as previously reported. And you can see that you turn on perfectly the peroxisomal protein in the cytoplasm. So we felt pretty good about that because, again, Heisenberg uncertainty principle was like, you always do not want to basically sort of disturbing the very measurement of the thing that you want to learn. Okay. Now, what's cool about this is that this locus is actually facultative heterochromatin. So when it goes, so in the presence of doxycycline, basically it has HP1 beta initially, sort of that will dissipate, and it's also encased in a PML body, which is great interest to me as a virus as well. And then again, sort of that begins to open up. So now we can do something really cool, which is iron has a different density, sort of in the periodic table than osmium. We can kind of have two color EM now, effectively, right? Where we basically stain the chromatin with our chrome EMT, okay? But now basically, could we see individual nanoparticles and find basically the actual gene structure and visualize a gene in the active and inactive state for the very first time? So that's what we did. We took the fire nano plus or minus dox, and then we did chrome EMT, okay? So, and here's the silent locus. Now remember, there's 200 copies of the transgene, right, integrated into one place, okay? I want to be clear about that. And each of these dots, took so long to work. I thought it was April Fool. I thought it was a Sharpie marker at the time. They were just playing with me, but it's not. So I've gone into a single slice now. So each one of these is actually your 8 nanometer iron core, basically, and it marks actually a lac GFP that's bound to the DNA. So you're actually seeing sort of individual sequences and the DNA. And this isn't the silent case. So now what we can do is we can effectively mark sort of lac sequences through this, both fluorescently and here. We're taking a trip through it. And I'll just turn up the contrast view so you can see it. And then I'll show you what it all sort of begins to mean. Oops, sorry. Go back a second. Yeah. And so I'll color in red now, basically the fire nanoparticles for you. Sorry. Manually. There we go. There they are, right? And so now we can see all this, and there's the chromatin. You start to begin to see this haze. If you look at it long enough, you'll see the Death Star. And so basically it forms this remarkable sort of a ball, right? And so now the question is, what happens when it's active, right? Same sequence, same everything. What happens? Does it change structure? And by the way, here's the scale bar. These are not higher order fibers, okay? You can see that for yourself, right? So here's the active locus, and there's a whole ton of things that I want to discuss with you here. So let me just play a few first. So now basically we've turned it on. And people had thought that the expansion of the locus initially reflected sort of an unfolding of a hierarchical fiber. Actually, it reflects a change in actually the concentration volume and actually the three-dimensional organization and the interchromatin distances actually between the polymers actually. And so now basically this is the lac sequence, and there's some really interesting other things that are going on, okay? But let me just take the first question. So is there basically higher order fibers basically in here, right? So here it is, and we can now just measure through the entire sort of volume. In general, basically, the answer is no. Again, mostly it's 5 to 24 nanometers. Where you actually start to see, I'll show you active transcription, you'll actually see an unfurling. But for the most part, again, we're not going from these hierarchical fibers into actually sort of that the primary polymer structure is mostly disordered or heteromorphic. Okay, but there are beautiful branches and structures. And then, but there's something really super cool that we can do is that about 40% of the genome is almost viral, right? It's actually repetitive. But how do you actually see repetitive gene sequences, even by genome engineering or not? What we can actually do is sort of statistical clustering, because I know that each LAC transgene has 256 LAC repeats. And so I can actually identify individual genes because I have 1 nanometer isotropic resolution. Okay. And so when you do that, you suddenly see the LAC sequences cluster here. And so they begin to define actually each individual transgene. So we can actually begin now to see the 3D organization of a repetitive locus, right? So now I'm here, now I'm projecting basically the chromatin structure underneath it. And then I can basically, by inference, here's the other sequences basically, here's the tetramotor, here's these other sequences. And I can begin to put them together. And you'll see actually a dramatic sort of a rearrangement of these actually. And so, and I'll try and explain it in the next piece. And so this is us modeling it here. So basically, this is the LAC part of the gene, and this is the sort of the TET kind of operator. And it's amazing actually the way they're actually arranged basically into this this pole. It's actually it's quite uniform actually. But what happens, as I'll show you when it gets activated, is actually basically some of the TET genes, it actually gets sort of, it's not activated all at the same time, they don't all open up. They actually begin to open up at the outside, but that actually pulls basically on the other genes. And now you actually get this 3-dimensional rearrangement of other genes that actually begin to go to the inside, which again, until I saw it, I was like, oh yeah, cool, right? That would actually have this effect in 3 dimensions, basically, where suddenly it starts to do this actually, right? And it starts to push some of these other sequences again into the center, which is really interesting in thinking about actually multi-copy genes, which are viral, they're translocations in cancer, and beginning to think actually about how actually these different mechanisms may play out. What's also really interesting is what happens is there's a critical change in chromatin concentration, where effectively this is basically border at about 35%. And then actually what happens is you'll actually start to see this get into about 12 to 20%, but you'll actually see some denser cores beginning at the center because this becomes self-inactivating at the outset, right? So that's the critical change that's occurring, was actually the concentration starts to dissolve kind of from the outside in, and that can actually create basically individual structures on the inside. And that I think is very key for then understanding RNA polymerase. So here's Pol2, and here's the locus here. And when it gets activated, you start to see RNA Pol2 beginning to gain access and to invade into it, right? So where's Pol2? Where's the RNA? Can we see that? Can we begin to see all of these elements? And so in the active locus, I said to you that there's lots of other things going on. Only in the active locus do we see basically the appearance of these 30 to 40 nanometer polymers, and these are just osmium densities. And these actually big densities, which always occur at the periphery, basically. Only ever see it when you get activation. So it's a function of gene transcription. And these amazing factories that appear right at the periphery, basically, upon gene transcription. So what are these? And so one of the things is that could these be sort of mRNP assemblies or even RNA DNA hybrids, which I won't get into, but that actually could represent potentially, as you're getting massive amounts of RNA being transcribed, very different to a single copy gene. It's actually a central feature by the way of an oncogene, as well as actually viruses. Maybe again, this is what you detect is the lack, that change in homeostasis, because that concentration then drives basically these interesting densities. And now Rob, in his wisdom, had put an MS2 basically binding site into the RNA. So we just used MCP GFP, and we use Fire Nano now just to see the RNA, right? So we can use the Fire Nano now to mark basically the RNA, the GFP tagged RNA. And what's really, really cool is most of the MS2 starts to accumulate actually around these densities. The RNA is more accessible, so labeling, even though this is the same basic affinity for GFP, labeling density is greater. So each of these represents a single cluster. You get about 18. So you're seeing individual RNAs that actually start to accumulate and pile up in these amazing factories at the periphery that we can see at sort of this resolution. And then one of these particles basically, so here's sort of this amorphous density in between, which is very interesting actually. These are basically the MS2s, but these are the particles. And actually, when we actually superimpose them, the mRNPs are associated right on top of basically the Fire Nano associated RNA, right? So that's why we think that they're basically RNA associated sorts of particles, which may actually reflect either the spliceosome or the mRNP, basically. But we can kind of get to that kind of a resolution now. Now, our first guess was hoping that these might actually be RNA polymerase, but they're not. And so the reason, so this is sort of, again, just to summarize where we're at. You go from dead star, basically, where it starts to open up the periphery. The RNAs get transcribed, high concentrations. They form condensates. And then actually you get sort of RNPs basically beginning to associate with the MS2 RNAs that occur there. The lac and tet basically fold out. And this is where you're starting to get basically the chromatin. Okay, what about RNA polymerase? So basically, we took GFP RNA polymerase. This is with Xavier Daszak. And this is alpha-mannitine resistant. This is basically FRAP experiments. So you can, because there's 200 transgenes, you see RNA polymerase there. So can we see RNA polymerase on a gene for the first time, right? And that's interesting to us. So here's Fire Nano now seeing just the RPB GF RNA. RPB is the major subunit, sorry, of RNA polymerase. Kind of interesting, again, to talk about protein-protein phase transitions. And obviously, being able to see the chromatin allows us to see the DNA polymer transitions as well. But can we now begin to sort of get a handle on both? This idea that at high concentrations of RNA polymerase, potentially as super enhancers and others, the CTD could associate through low affinity interactions to sort of drive sort of transcriptional initiation at a critical mass. Well, does it? We have 200 copies there. Where's RNA polymerase? So why don't we look? Okay. So here's again your chromatin, right? Basically in the density. And now basically each of these little black spots is actually an RNA polymerase, okay? So let's, so now that basically will actually mark basically the RNA polymerase, and you can see it basically sitting on top of the DNA. And it's actually several things that are really cool, but let's just take a trip through the locus. There's a little bit of a cluster here, I'll come back to it, but mostly actually they're uniformly distributed, which was actually very much predicted actually. By Ibrahim's, Xavier's, Rob's sorts of experiments of the years in terms of actually sort of statistically predicting it. Every now and then, let me see if I can find you one. See these little two? They're right next to each other. We think those actually might be right on the promoter because they're individually associated with DNA. Some of these that are out here just are diffusing away. They're actually in the middle of space. But every now and then, they're actually a cluster. But here's our 30 nanometer particles. I was hoping that that would bind to that. It's not. We're still trying to figure out what this is. But here is the RNA polymerase. So now we can actually begin to build these things in. What's cool is we can actually kind of ask a key question is, when do you get clusters of RNA polymerase? Is there a critical number? Because we can actually count them. And it turns out, so far, basically, we can see 70, we can see 15. 15 is the minimal so far. But there's something really interesting that when we draw the distance between each of the RNA polymerases, it's not random, in fact. It's not at molecular distance either. But effectively, the distribution always falls between about 15 to 30 nanometers. And the CTD of RNA polymerase is 16 nanometers, if stretched out, which is the least entropically favorable structure, of course, that it could adapt. But it's actually about 15 to 30 nanometers, which would be consistent potentially with the CTD mediated interaction. So we think that there might be a critical number such that we can measure, but also a critical distance that is consistent with it. I'll just end with sort of answering a key question, is that people will draw some of these sorts of protein condensates as kind of being driving, kind of binding into each other. But with the case of RNA polymerase, that would actually potentially inactivate it, right? Because it would actually take it away kind of from the DNA, right? It's supposed to actually zip along the DNA. But could it actually be then associated with chromatin? How could that work, right? Which is it, right? So we can actually answer that question because we can actually look. And so that's what you're seeing here. So we just took the yeast Pol 2. And again, I'm not modeling, I'm not modeling helices before anyone screams at me. I'm just taking just the general density and shape to see if it's consistent. And we can take each of these spots here and look at the chromatin and say, is it on chromatin or not? Okay. Or are they actually intimately associated with each other in 3D? And you'll see that here. So let's look at that cluster. This is very near to that other one. So we drew these in actually. What you'll see is actually that density was very gratifying. Is the Fire Nano was actually exactly the right distance away from that and the chromatin, which you'd expect because it's binding to it. What you can see is that these are actually associated with chromatin. So potentially it can actually stretch across potentially these structures as they mount up and mediate that with the CTD and potentially these gossamer-like interactions perhaps. can actually mediate these densities, which I kind of thought was a bit of a conundrum, actually, right? So you can have potentially polymer-polymer interactions and protein-polymer, protein-protein interactions coexisting. And that may differ depending, again, on whether you're dealing with activation versus repression. So my last slide is just to sort of, two things I want to leave with you is that, is there a general sort of a principle where chromatin volume concentration, which could be mediated through different devices, polymer-polymer, protein, and other interaction spatial, that could actually sort of determine whether it can be accessed by machines such as the spliceosome and other. This is sort of a somatic cell. This is our heterochromatic locus, where basically it's kind of going from here to the active. That's what I just showed you. So it's consistent with that model. We've also been looking at human embryonic stem cells and differentiated cells, and these sort of seem to be more generally open and sort of exist on this continuum. So, and also kind of polycomb bodies and other things. So right now it's generally consistent at this scale. I have no idea at a single gene level. I do want to be very careful to say that. At the scale, this is consistent and I think intriguing. This allows us now, I think, to really go after sort of dynamic chromatin structures in a really interesting way. And so what you saw was basically sort of a multi-copy transgene. It gets activated actually from the outside in, and it starts to unfold kind of here. High concentration of RNA basically drive then condensates at the periphery and the association of these particles. Whether these are activating or inactivating remains to be determined. And you'll actually see RNA polymerase and some clusters sort of beginning to kind of pile up here. Then you also will rearrange the 3D chromatin structure of tandem genes and start to create some really interesting effects as you pull on them. And so it changes your ideas about how we begin to think of newly evolving genes, right? Because exonization drives gene structure and all these different things. I'll leave you with this final slide because I will, on an open call, is that, we have all these data sets, but how on earth we actually analyze them basically, computationally? I need help. I would love help because there's just so much information in them, and how we statistically describe this, I think, is obviously sort of right in Vadim's wheelhouse. So this is a completely sort of, selfish kind of call going, please help. But one thing I'll leave you with, because this is actually U2S cells, which is an alt-driven cancer, a deadly cancer actually in children. And this is this lac locus that recombines basically, and this is again how you get amplicon. I just think this is really interesting. So that's this lac transgene now, and it's gone into another locus, and it's multi-copy basically. But what is amazing is here it is. Oops, just want to play it too. This is kind of a Death Star-like effect. Is that while this is not the same structure, right? There's actually more repeats in this, as what I just showed you. Even by eye, I can see that this is statistically similar, right? Again, it forms this kind of almost Death Star-like structure, becomes more inactive. There's actually more volume of DNA, harder to activate. And it really does remind me of Luke Skywalker, right? And it's more separated out basically. So the copy number has increased. And again, it's gone into this other locus, but at that particular sort of, I'm not sure if it's a critical genomic mass or in situ mass that can actually drive sort of now either polymer-polymer, protein-protein driven interactions that can actually lead to a similar sort of structure in a completely different place with potentially kind of a different context. And I think that's kind of interesting and really relevant for how viruses integrate, because they tend to do it as multi-copies. Amplicons, EGFR, glioblastoma, that's how we get drug resistance. And so again, can we begin to understand this? Which is probably fundamentally driven by polymer-polymer and these protein low affinity interactions, to begin in my ultimate dream to realize sort of drugs for diseases that there's no hope for. So you're the hope. So with that, let me thank everyone who contributed in my lab. This is an amazing collaboration with Mark's lab. Get into the Fire Nano, Horn with the Chromium T, and yeah, I'm happy to take any questions, or I'll go to the panel at the end. But. You're welcome. Okay.
I have one word. Wow, this was amazing. Thank you so much. All right, I would like to introduce let's see. My good friend Hao Zhang. He is also a member of the Physical Genomics Center. He is a professor of biomedical engineering, internationally recognized for his development of optical microscopy and for acoustic microscopy, and he will introduce the next speaker.
Thanks. Hi, I'm Hao. I'm a member of the Genomics Center and also co-PI of the U54 grant. It's my pleasure to introduce the next speaker, Dr. Karpen. Dr. Karpen received a PhD from Washington in 1987 and performed the postdoc research with Dr. Allen at the Carnegie Institute. And then he became a faculty member at Salk Institute. Later on, moved to Lawrence Berkeley National Lab and part of the UC Berkeley campus. Dr. Karpen was a Pew Scholar in the biomedical science and has authored numerous number of high impact papers, which I cannot read. Otherwise, we'll go to lunch directly as well. The research interest of Dr. Karpen focus on long-standing chromatin structure and function correlation with a special emphasis on the impact of chromatin inheritance, nuclear architecture, genome stability, and human health. I can't make a very interesting introduction like Vadim did, but seeing that picture remind me during the winter a few months ago, my family tried to escape the Chicago winter. We spent a week in San Diego. My wife saw this picture from many news articles and asked me whether we can go there and take a picture. So we drove to Salk. Of course, we were blocked, saying that, no, you need a host. Like, I look at my contact number. I don't know anyone there. So we took a picture outside. It can't go in there. Without further ado, let's welcome our next speaker, Dr. Karpen.
I got this one. Let's see. Found it. Give me a minute here to get. Please. I think we're not hooked up to Zoom. Hold on. Sorry. I don't know what happened to the. What happened to our AV guy? Mhm. There we go. Is everything cool with the? I gotta go. Yeah. Video panel. All right. So thank you so much for the invitation to come to this symposium. It's really exciting. I mean, Claudia's talk was amazing, and I'm sure the rest of the talks will be as well. I'm very excited to be here, despite the fact that I had to miss the first Warriors playoff home game last night, but they won anyway, so I'm happy. All right, so my speaker is on. That's not good. All right, so I actually have a different title. Decided to talk about something a little different today, and so I'm going to talk about dances with nuclear condensates and the interplay between three different condensates, heterochromatin nucleoli, and a new one that we've discovered called Sheila. All right, so Clodagh gave an amazing introduction to nuclear architecture. I'll just remind you about the focus here, and that is that nuclei have numerous bodies, each of which have distinct components, distinct functions, and distinct structures. And so the two most prominent ones are heterochromatin and nucleoli. And those will be two of the things that I'm going to focus on today. They generally constitute about 2/3 of the volume of the nucleus. But there are many, many other bodies. Other chromatin states, such as polycomb, are associated with bodies. Different functions, such as DNA repair, replication, transcription, telomeres, splicing, et cetera, are all associated with bodies that have distinct components that are associated with the functions that they encode. Now, looking at these types of pictures makes me wonder whether there really is such a thing as nucleoplasm and whether the nucleus is really just filled with these distinct bodies. Now, most, if not all, so all of these are membraneless. The only membrane in this picture is the nuclear membrane. All of them are membraneless, and most, if not all of them, behave as liquid-like, what we like to call biocondensates. And Clodagh gave you an introduction to phase separation. Likely that these are formed by phase separation or similar mechanisms. Now, I recognize that using the term phase separation is somewhat controversial at this point. And I think that it's good to hark back to Schrödinger, who in his book, What is Life, where he basically said, well, biology follows the natural laws or the laws of physics. And I'm going to paraphrase here, but there's a lot of shit loaded on top of it that obscures the fundamental physical laws. And I think that that is one of the issues here. We can see phase separation happening, whether it's liquid-liquid, liquid-gel, whatever, in vitro, very simple systems. It's much harder to prove in vivo because you can't generate the same kind of phase diagrams that you can do with purified components in vitro. However, you can see all the signs of liquidity, molecular mobility, et cetera, et cetera. I think more importantly that the question is not whether phase separation happens in vivo or not, but we have all these bodies. They behave in many cases as liquid-like entities. How are they formed? How are they maintained? How are they dissolved? How do they impact genome and nuclear organization and function? To me, those are the most important questions. These are entities that we need to understand. And that's what we're trying to do, along with many others in the field. Now, we spend a lot of time working on the heterochromatin. I'll give you a little bit of a flavor for that. But what we've started to think about is how do these different condensates interact both structurally and functionally? So what is the interplay at both the structural and functional level between these condensates? And that's what I'm going to talk about today. Very much work in progress, but I hope this will generate some discussion and some thinking on your part as well as mine. All right, so our favorite condensate is the pericentromeric heterochromatin. And I want to make a distinction, which is that the term heterochromatin is often used now, especially in the genomics field, to refer to any silenced locus in the genome. And that's not what we're talking about here. Although there are commonalities, what we're talking about here is the fact that, and here's the Drosophila karyotype, but the same is true for mammals, including us, and that is that around the centromeres, the region where the kinetochore forms, it's responsible for attachment to the spindle and chromosome segregation, there are megabases of highly repeated sequences, so-called satellite DNAs, simple sequences like AAGAG, repeated for hundreds of kilobases, up to megabases, and also transposable element fragments. These surround the centromere on all chromosomes. So there's very large domains, and that's distinct, and also at the telomeres. That's distinct from much smaller regions of silent chromatin, which might have the same composition in terms of histone modifications and reader proteins and things like that. But those are much smaller regions. Okay, now even though the pericentromeric genomic regions are separated in terms of being on different chromosomes, if you look at the interphase nucleus, they coalesce to form what are called chromocenters. And so the organization of the nucleus reflects their ability to come together in three-dimensional space and form clusters, as we saw in. All right, so pericentromeric heterochromatin, or PCH, is what I'll be calling it, are liquid-like condensates that are associated with repeated DNAs. There are few genes in the pericentromeric heterochromatin, but there are in fact genes there. In Drosophila, for example, the only copy of MAP kinase and the only copy of the RAD21 cohesin cleasin are actually embedded in this so-called silent region, and these are essential genes. So there are genes there, but for the most part, what the pericentromeric heterochromatin does is mediate chromosome segregation, things like meiotic pairing, obviously centromere function, sister cohesion, and silencing of genes and transposable elements. We, along with Gita Narlakar's lab a few years ago, no, I guess 6 years ago, showed that the pericentromeric heterochromatin behaves as a liquid-like condensate. And I'm not going to go through all of the evidence that we presented then. Excuse me. But the bottom line is that we spent a lot of our effort looking in vivo. Gita spent a lot of her effort looking in vitro. And so both in vitro and in vitro, we could see all the properties of liquid-liquid phase separation reflected in this condensate. Now, the mechanism, the molecular mechanism, is that these repeated sequences are highly enriched for one particular histone modification, histone H3, lysine 9, di and trimethylation. And its associated reader protein, epigenetic reader protein, a heterochromatin protein 1 or HP1, which contains a pocket called the chromodomain that specifically and uniquely binds to di or trimethylated histone H3 lysine 9. Now, I'm not going to go into the details. We have a lot of work that we've done focused on how these form in vivo. And basically, yes, you have recruitment of HP1 to K9 methylated chromatin through this chromodomain. But in reality, it's this giant hairball of interactions. That is, HP1 exists predominantly as a dimer, and it interacts not only with itself and with the chromatin, but it also oligomerizes, so that oligomers of dimers. It also interacts with DNA or RNA through disordered domains. It, at the dimerization cleft, has the ability to bind hundreds of different binding patterns, each of which are themselves multivalent and contain disordered domains and other structured domains that mediate weak hydrophobic or electrostatic interactions. So it's this giant hairball of interactions held together by weak multivalent interactions. All right, so I did want to stop before going, or pause, not stop, before going into today's topic and try to at least give you some sense of why I'm excited about thinking about the role of condensates in chromatin biology. And that is because it raises new possibilities for thinking about regulation and for both in terms of organization and things like gene activation or DNA replication and things like that. And I think this is really important in the context of thinking about engineering, not just genome engineering, but how we engineer the nucleus, that we need to be thinking about something beyond how genome regulation may involve open versus closed chromatin. This is really pervasive in the field, that either you have open chromatin or closed chromatin. I think Clodagh showed very nicely how there's much more complexity there with the addition of the polymer. But I think if we think about it, liquidity can have major impacts on genome organization, such as the ability to form hubs or compartments from self-fusion. So when you have condensates of like composition, they can undergo liquid-like fusions. If they don't have the same composition, they won't fuse. HP1 condensates will fuse with each other. HP1 condensates will not fuse with polycomb condensates because they don't have a molecular compatibility. And so, for example, we had shown a few years ago, Grace Lee, who was at the time a postdoc in the lab, that if you look at transposable elements strewn throughout the euchromatin, they are actually small regions of K9 methylation associated with HP1, but they, in 3D space by high C and by FISH, actually associate with the pericentromeric heterochromatin in this manner. Another possibility, which we're currently testing, is that instead of thinking about the spreading of chromatin states, which is a well-known property of the heterochromatin, as a processive enzymatic reaction, which people like Jerry Krabte have essentially ruled out based on the kinetics, is that maybe spreading of chromatin states is more of a wetting process, again, reflecting. The other thing is that instead of thinking about chromatin compaction, that is the inability of things like Pol II to actually enter into a region because the chromatin itself, the DNA nucleosome polymer, is compacted. Once you start to invoke condensates, you can think about the biophysical properties of the condensate as a whole, such as the ability to selectively exclude or retain genome regulators independent of the packing of the chromatin fiber underneath that. And then finally, from work that we did on DNA repair, where we see that double-strand breaks made inside the heterochromatin domain, so this is HP1, shown here in red, and these are repair foci arising after irradiation. We showed that, in fact, these foci have the double-strand breaks have to move outside the heterochromatin domain and actually go to the nuclear periphery to complete homologous recombination repair. And although Aninicolo and others have shown that once they leave the heterochromatin domain, they move to the periphery due to actin-myosin-mediated directed movements, that is not the case in terms of how they leave the domain. And so one of the things that we've explored is the idea that, and I think Andy has actually worked in this area, and that is this behaves as that the DNA nucleosome polymer is behaving as a copolymer with multiple different epigenetic states all along it, and that you can really understand how different parts of the polymer can move in and out simply based on the energetics of the ability of, for example, if you are K9 methylated, you will interact with HP1, and if, as we see in all these different, we've described this in different publications, during repair, the initial stage of repair, K9 methylation is removed, that part of the polymer can no longer interact favorably, interact in terms of the energetics with the heterochromatin domain and will essentially ooze its way out. So again, much of this is fantasy. We're testing a lot of these ideas. In fact, if we go in and directly demethylate a region of the heterochromatin, we can actually see it moving out of the heterochromatin domain. But these are all works in progress, but I think what I'm hoping is that you get an idea that it's much more than just potentially much more than open and closed chromatin, and that we need to come to grips with that. Okay, so this is the theme today. I'm going to talk about hopefully interplay among three condensates if I don't run out of time. So first we have the pericentromeric heterochromatin that's shown in pink and nucleoli shown in green. And then we have a new condensate, which we call Shelob, and you'll see why, that actually is present in all the interchromatin spaces outside in the euchromatic space, but also surrounds the heterochromatin, the pericentromeric heterochromatin. So there's like this three-layered structure. And so we want to understand how is this organization established, and are the associations important for organization functions. And again, this is all new, unpublished, works in progress. So this is, let's first focus on nucleoli and the pericentromeric heterochromatin. This is a highly conserved organization. You see it in human nuclei. So here's the heterochromatin surrounding nucleoli. You see it in Drosophila, plants, et cetera, et cetera. The work I'm going to talk about in Drosophila was done by a really talented postdoc, Barsha Rashekar. And so I told you about what the pericentromeric heterochromatin is. Nucleoli, as I'm sure many of you are familiar with, is a multi-layered condensate, as first shown by Cliff Brangwyn when he was a postdoc in Tony Hyman's lab. And the key to understanding nucleoli is that they're built on another repeated genomic element, the ribosomal DNA. These are much larger repeats, depending on the organism. In humans, there are 45 kilobase repeats, but there's hundreds of copies located on different chromosomes. But the key to understanding nucleolar formation is that it's transcribing ribosomal DNA. So for the formation of ribosomal RNA from RNA using RNA pol 1, that actually creates the initial layer. And this is called the fibrillar center. And then there's a second layer that's formed by the recruitment of processing factors, things that will process the ribosomal RNA, such as one that I'll focus on, which is fibrillarin. And then in the outer layer, you have what's called the granular component, which is where ribosomes are being assembled. And each of these, like in the case of all the different condensates within the nucleus, really reflect different condensates or different phases. They have different composition, different sets of molecular interactions. So this is the site of ribosome production. Now, in flies, in humans, our DNA is embedded in the pericentromeric heterochromatin. And so you might imagine that this association is simply because of genomic proximity. But it turns out, so here's flies, there's about 200 repeats each of the rDNA on the X and the Y. but in fact, none on the second or third or fourth chromosomes. And yet they're also surrounding the nucleolus. Okay, so it's not simply because you are you have a physical association in cis that you have this surrounded configuration. All right, so what Varsha did was to look at the initial establishment of nucleolar and pericentromeric heterochromatin condensates in early Drosophila embryos. This is really a wonderful system because you can look at the very first stages of formation of these bodies and not worry about whether you're just looking at a maintenance stage as in cultured cells. So early Drosophila embryos, you have fertilization, and you have a bunch of nuclear cycles. You don't get cellularization until cycle 14, and then you have gastrulation and you make a larva. What's interesting about these cycles is that they're just SM cycles, or DNA replication, mitosis, DNA replication, mitosis. And these interphases, especially early on, are incredibly short. They're 6 minutes to start with, 15 minutes in cycle 13, and 90 minutes in the first more extended interphase in cycle 14. Now, heterochromatin foci, as Amy Strum in my lab first appear in cycle 11. You need to reach work that I'm not going to talk about. You have to achieve a certain concentration of K9 methylation on the chromatin. That requires this many cycles. That's when heterochromatin foci first appear. Nucleolar foci, as shown in the Wieshouse lab, first appear in cycle 14, so 2 cycles later. So 13 is the first time when you see both. And so what we're going to be looking at is a bunch of movies where we're marking the heterochromatin with HP1, RFP, and the nucleolus with fibrillarin GFP. Now, when they start out in cycle 13, they are not in this surrounded configuration. And by later in development, they are. And so the question is, how do you get from here to here? And so one important point to make is that in every single one of these mitoses, both condensates dissolve. All condensates that we know of dissolve in mitosis. So in every interphase, they have to redo this process of starting out with all the molecules being diffuse, then nucleating, and then growing. Now, in these early cycles, as you'll see here in cycle 13, there's only 15 minutes in interphase, and so they don't have a lot of time to grow, and they don't undergo fusions. And so this is what you're going to see. I guess there's a little too much light, but you can see that they come out of the previous mitosis, they're diffuse, and then heterochromatin bodies first appear, then nucleolar bodies appear because ribosomal RNA is starting to transcribe. But they remain separated in space, and there's not enough time for them to grow and undergo liquid-like fusions. In the next cycle, in cycle 14, you see something very different. They start out exactly the same as they did in cycle 13, but they now have 90 minutes to grow and fuse. And so that's what you're going to see here. They're now growing much larger, the green, the nucleolar components, HP1 and fuchsia. And you can see them, again, creating much larger, here's a much larger volume, occupying much larger volume of the nucleus. This is the side view. Bottom line is that at this stage, by the time you get to the end of cycle 14, you're still not in this surrounded configuration, this canonical, highly conserved configuration. You have what is a more elongated branch structure for HP1 juxtaposed with the nuclear periphery, the nuclear lamina. And then there's one part of the heterochromatin, which corresponds to the sex chromosomes, the rDNA locus, which is in fact sort of inverted. That's surrounded by the nucleolar material. But the important thing is that if you now look at subsequent cycles, I'm not going to dwell on this, I want to get to the functional studies, is that what you see is that in cycle 15 and 16, you're still in this elongated configuration. In cycle 16, it starts flipping back and forth between the association with the nuclear lamina and the surrounded configuration. And it's only in cycle 17 that you now get a stable surrounded configuration. Okay. So the bottom line here is that this canonical organization is assembled by a very dynamic process. It just doesn't appear de novo. There needs to be a bunch of stage-specific intermediates to get from this point to this point. I didn't get into it, but there's a lot of interesting, I think, biophysics to think about because of changes in nuclear volume and changes in the speed with which this kind of like with which these different structural changes occur becomes shorter and shorter and shorter in each cycle. So we don't understand that, but we're trying to figure that out. All right, mechanistically, what could be happening? Tons of things we don't understand. So it could be changes, as I said, relative volumes of the two condensates, aspects of the nuclear volume changing, wetting of two immiscible liquids, et cetera, et cetera. Lots of different possibilities we're investigating with different types of experiments. We've also, and I'll come back to this, but just as a hint, we've been working with Sam Safran's group at the Weizmann Institute, integrating physics and different types of simulations. And this has led us to this idea to favor this idea of this being formed through some kind of affinity hierarchy, just solely based on interactions between the different molecules. Before I do that, I want to turn to the question of, is this, this has been descriptive. Here's what the dynamics are. I really want to talk about this, not because that's particularly interesting, but because I think it's really important to keep these dynamics in your head and understand what's happening live. But first, I wanted to turn to whether manipulating one condensate or the other impacts the ability of manipulating one of the condensates impacts the organization of the other. And I'm not going to go into details of when we get rid of the heterochromatin, the nucleoli become bigger, you get much more ribosomal RNA. We think that this is not an interaction between condensates per se, it's because most of the rDNA is silent, and it's silenced by K9 methylation and HP1. So if you get rid of K9 methylation globally and you reduce the amount of heterochromatin, you get a much bigger nucleolus simply because it's transcribing more rRNA. It's possible there is interaction there. What was more interesting came out of looking at what happens when you get rid of the rDNA. And this was done by the Wieshouse lab back in 2017 using a what's called a compound chromosome, a highly rearranged chromosome in Drosophila, something you can do with Drosophila genetics. And basically you can create embryos that have no ribosomal DNA at all. Now these animals live till late embryos simply because the mother sticks in tons of ribosomes into the egg, okay? But the important point is that all these stages that we're looking at when you normally form the heterochromatin and the nucleoli, they're perfectly normal in terms of timing and all that sort of stuff. So they're living high and they're living well off mom's ribosomes. All right, so what happens when you get rid of nucleoli? And there's a number of really interesting phenotypes, and they raise a lot of important points and questions. The bottom line is, here's plus rDNA, you have the nucleolus, you have the heterochromatin. This is cycle 14, it doesn't yet surround the nucleolus. Here's what happens in the minus rDNA case. First and foremost, fibrillarin, this nucleolar component, has nowhere to nucleate. There's no rDNA, there's no rRNA. So it actually goes off because it has inherent self-association and forms what we call a neocondensate. It forms this other beautifully spherical object, but not connected to the heterochromatin. And that's what you see quantitated here. Other nucleolar components, like this outer nucleolar component module, just gets dispersed throughout the nucleus. And that's because it doesn't have self-association. You'll see there's a third pattern, abnormal pattern that happens. All right, so that's one phenotype. The second phenotype that we were very excited about is that, in fact, the pericentromeric heterochromatin becomes abnormal. And it becomes hypercondensed. You don't see this elongated configuration you see in wild type. It's much more condensed. You can measure this by looking at the aspect ratio. And so you can see in the minus rDNA case, it's significantly more condensed than in the plus rDNA case. And if you do fish with different satellite sequences, you can see in wild type, they're much more dispersed throughout this volume, and they're much more compacted in the absence of the rDNA. So what we conclude from this is that nucleoli actually are required to regulate the distribution of the pericentromeric heterochromatin in 3D space, and especially to keep the heterochromatin from hypercompaction. We can discuss later about, is that because of an inherent property of the polymer that's K9 methylated plus the HP1, but there is this ability to become more compacted if you don't have this nucleolus to assemble around. All right. There was another phenotype that really turned out to change how we thought about this process in fundamental ways. And that was this. In subsequent cycles, in cycle 17, which happens to correspond to the cycle when you get this surrounding configuration, what we saw was the formation of not this, within this compacted pericentromeric heterochromatin, so this is the minus rDNA case, we see this mysterious void appearing, a black hole or whatever. And you can see this in this movie. So here's the fibrillarin neocondensate, and you can see down here and up here, you can see as this cycle 17 moves forward, this void appears. And you know, for a while it was like, oh, that's sort of interesting. But what's really interesting are these neocondensates that form. That's now this persists throughout embryogenesis. You see this hole here. And so, yeah, what's in there? So Varsha went and looked, you know, all sorts of candidates. It's not RNA. There's no DNA in there that we can find, or at least not high concentrations, no histones, no nucleolar components, a whole list of things that aren't there. What is there is protein, and this is just showing a pan-protein label using the 488 NHS ester. Here's the void that's formed, and then here's a high concentration of protein. It looks a lot like what happens in the post-rDNA, where there's one of the highest concentrations of proteins in the nucleus is in the nucleolus, but that's not a nucleolus. The fibrillarin, remember, is somewhere off to the side. All right, so what are these proteins, and what can they tell us about nucleolar heterochromatin assembly? So this is where our collaboration with the Safran lab really paid off. And so in working with Omar and Gaurav and Sam, we were thinking about, okay, what kind of proteins could be involved in both assembling the heterochromatin around the nucleolus and in creating this void in the abnormal case where you lack the rDNA. And so we thought about it and came up with this idea that, well, maybe they're amphiphilic proteins, of course, your standard protein X. And the properties that it should have is that it needs to self-associate, and most importantly, have affinity for both the nucleolus and the pericentromeric heterochromatin. Okay. Pretty simple, but it took us a while to get there. And so the idea is that this protein X would be able to sort of glue or stabilize the association between the pericentromeric heterochromatin and the pericentromeric heterochromatin in the nucleolus. And in the case where you have no rDNA, due to its self-association and its affinity for HP1 pericentromeric heterochromatin, would form or fill this void. And you know, this is would be another type of neocondensate, right? It's an abnormal structure that forms because you've lost your primary nucleation site. And now proteins that are used to undergoing weak interactions with other proteins or nucleic acids will now go off and find another place to accumulate. And so with Sam, Omar, and Gaurav, well, they did this. They developed a 4 minimal component coarse grain model to help us think about this. And it's very simple, and I'm not going to go into great detail here, don't have the time. But basically, we have the heterochromatin as a DNA nucleosome polymer. Embedded in that is the rDNA, our RNA. Remember, this is all coarse grain. Fibrillarin as just a representative of the nucleolus, and then this protein X, this amphiphilic protein shown here in yellow. And the bottom line is that we set up an affinity hierarchy. And we decided that this was based on known properties. Others were guesses. Bottom line is that we tested a range of affinities. We didn't just tune this so we got to get the result that we wanted to get. I'm not going to go into detail for this, but basically this type of hierarchy fits what we know about these proteins and about these condensates. And so if you run the simulations, you end up being able to recapitulate the surrounded configuration, again, over a range of affinities, not just by tuning it to one or the other. And I think most importantly, we were able to also show that using the same modeling, that if you remove the rDNA, and therefore you get rid of these affinities, okay, the ability of rDNA to interact with fibrillarin, as a nucleolar component, fibrillarin's interaction with this magical protein X, then in fact you can recapitulate again over a range of affinities the Fib neocondensates, and this void that is filled with this protein X. Okay, all this is well and good. That just tells you what's possible. It doesn't tell you what happens. And so, is there a protein X? And if so, what is it? And we got very lucky. And I'll tell you why we started out picking this protein. We got lucky and identified this protein pitchun, which is a dead box RNA helicase. And the reason that we picked this protein, which not a lot is known about in flies, there is a human homolog called DDX18. Number one, this protein has nucleolar affinity. Okay, it sits in the right place. It's in the outer layer of the nucleolus. It's not just at the surface. It fills the whole outer layer of the nucleolus. And so you can see that here. Here's fibrillary in pink. So that's a more central component. And then this is this pitchun component around the edge. So it fulfilled one of our criteria. It has affinity for the nucleolus. Second is that it also has affinity for the pericentromeric heterochromatin. It contains what's called the PXVXL motif, which is a motif that is necessary for interacting with the dimerization cleft of HP1. So many of the most of the hundreds of HP1 binding partners contain this PXVXL motif. Okay, and then secondly, from the work from the Wieshouse lab, they didn't look at HP1, but they noticed that in the -R-DNA embryos, pitchun accumulated in this body, an abnormal body, which looked a lot like Barsha's images of what the HP1 looks like in the -R-DNA embryos. Okay, that was suggestive, but certainly not proof. And the bottom line is that yes, obviously I wouldn't be talking about this if this wasn't true, but it turns out that yes, pitchun is not only located in the outer part of the nucleolus, juxtaposed with HP1, but in the in wild type, but in the -R-DNA case, it fills this void. Okay, so this is the HP1 here, and this is this pitchun protein tagged here. So you can see that in all these different embryonic nuclei, that's true. And I think you can see this in movies. And so you can see this is again the cycle 17. It starts out where the HP1 is in this condensed state. And then as the cycle proceeds, pitchun gets more and more and more pitchun gets recruited. And we think this is generating this void. Okay. So together with the affinities, it suggests, obviously does not prove, that pitchun is at least behaving like this protein X. And we think that it is likely mediating the layering of the nucleolus and the pericentromeric heterochromatin in wild type, and the formation of this void in the -R-DNA case. And so finally, can we show that if you get rid of pitchun, you actually lose this surrounded configuration? And the answer is a tentative yes. We did an RNAi experiment in S2 cells. Unfortunately, if you knock out, to get rid of pitchun in embryos, you have to first get rid of it in oogenesis. At least that's what we tried to do, and it doesn't make eggs, so you don't get any embryos. So it's actually essential for oogenesis. We'll get around that. So we did this in cultured cells, and in fact, what you see is what we would have predicted, which is that instead of this surrounded configuration, you have a much more compacted HP1. It surrounds less of the nucleolus, and that's exactly what we would have predicted. Now, from this, we tentatively conclude pitchun is required for the surrounded configuration. And the tentative part of this is this data from last week, and we need to do a lot more quantitation, and we have a lot more experiments to do in general. How much time do I have?
Good.
I know I'm good, but how much time do I have?
20 minutes.
20 minutes?
You want, we're ahead of schedule.
Well, let me start a whole different talk. No. All right, so then I have time for the last 10 minutes. There we go. All right, so from this part, the first 2/3, the conclusions are, number one, the formation of this surrounded configuration, this interaction between the condensates, is highly dynamic. And then we also, from getting rid of the rDNA, conclude that nucleoli are in some way regulating the distribution of the pericentromeric heterochromatin and prevents its hypercompaction. We think that based on the modeling as well as in vivo experiments, that there's a hierarchy of interaction strengths that underlie this surrounded configuration. And in particular, we've identified one protein. There may be others. We're looking for those, doing proximity labeling. But it seems that at least Chuan is at least a linker between the nucleolus and the pericentromeric heterochromatin and fills this void in the -R DNA. And I want to, I'm going to emphasize this again at the end, but I think one of the most important lessons, and maybe the one thing that I would ask you to remember from this, is that I think that what we're seeing with respect to manipulating these condensates individually and collectively is that there are enormous implications for mutant phenotypes that we need to consider. And this is based on seeing these neocondensates and mysterious black holes. If you lose the nucleation site for a condensate, remember it's based, the whole formation of these, at least liquid-like condensates, is based on weak multivalent interactions. If you lose the nucleation site or get rid of it, or you get rid of one domain in a multivalent protein that's responsible for interacting with other proteins and recruiting them into the condensate, then you need to keep in mind that the things that it's going to interact with, such as what we saw with fibrillary, you get rid of the rDNA, fibrillary goes off and does whatever it wants to do. In this case, it self-associates, but other proteins will go and associate with secondary sites or tertiary sites or quaternary sites. And so I think that's really important. And we've seen in this case so we have 3 different phenotypes associated with getting rid of the rDNA. Some proteins just get dispersed. Those could be neomorphic in terms of their effect on phenotype and disease. Some make neocondensates like fibrillary, they just go off and self-associate. And then the third was this Chuan behavior, which is that it now fills this void simply because it has both self-association and the ability to interact with HP1. All right. Now, the last part, I'm going to talk about this other condensate that surrounds the pericentromeric heterochromatin. And there are themes that are now coming together, and specifically this hierarchy of affinities. But as you'll see, we haven't quite put it all together yet. This is work from former graduate student Joel Swenson and a staff scientist, Serafin Colmenares. So we got into this protein because we identified it in an IP mass spec experiment using HP1 as bait. So it's an HP1 interactor based on that. We at the same time are doing an genome-wide RNAi screen looking for things that disrupted HP1 organization in the nucleus. And it was the only protein, like the Highlander, it was the only protein that came out of both screens. All right, I know that the Highlander reference is not, maybe 2 people here get that. Yeah, okay. All right, so AlphaFold is pretty useless for this. And the reason for that is that this is a 969 amino acid or 919 amino acid protein. It has 3 structured domains, zinc fingers. So there they are in the AlphaFold, and the rest of it is just flopping around in the wind. Bunch of disordered domains. It is a pain in the ass protein to work with. Do not ask me why we haven't done in vitro phase separation experiments, because you can't get the damn thing out of bacteria. Anyway, it has all these disordered domains as shown here, only these 3 zinc fingers. So it is a bona fide HP1 binding partner. In reciprocal IPs, if you pull down HP1 or you pull down Shelob, you will pull down the reciprocal. And like with Chuan, it contains one of these PXVXL domains here down in the C terminus. So it looks like your standard, biochemically identified binding partner, right? And most importantly, it's required for organismal viability. It's also required for forming the PCH condensate. So if you get rid of Shelob, this is in mutants or by RNAi, then you lose the HP1 condensate. So here it is in, sorry, that's very small, but in heterozygotes versus homozygous mutants, you also lose pericentromeric heterochromatin functions. You lose the ability to silence genes. This is a LAC-Z integrant that gets silenced. And then if you can alleviate silencing using what's called the Suvar mutation, that is mutating something required for heterochromatin formation. Shelob gives you also an increase in gene expression. It also alters if you mutate it, alters this, it's required in wild type for the spatial temporal dynamics DNA repair in heterochromatin. So it's impacts pericentromeric heterochromatin organizationally or structurally and functionally. But there's no change by ChIP-seq in HP1 genomic localization or protein levels if you do live studies or Western blots or IF. So it's very weird. Mostly that's a change. Those are changes you would see with something that affects the organization and function of heterochromatin. Even weirder, it's very different in its distribution. So most HP1 interactors, which we published back in 2016, either completely co-localize with HP1, here's HP1, here's an interacting protein, or they form these little subdomains inside the HP1 condensate, but not Shelob. By confocal imaging, what you see is that it has two patterns. It's not exclusively surrounding the heterochromatin. It has a web-like pattern that fills the interchromatid spaces. So sorry, here's Shelob. So out here, it's filling interchromatid space. I'm not showing you, if you stain for histone, they're mutually exclusive. So it's in the interchromatid spaces. That's why we named it Shelob, by the way, for the giant spider in the Lord of the Rings. So it's the web-like pattern, but it also has this enrichment, and it's rich in region very low histone density, and it's enriched around the pericentromeric heterochromatin. So Shelob is in red, and so you can see it doesn't fully surround it, and there's some other localizations, but it does show enrichments in parts of the pericentromeric heterochromatin. But it doesn't actually physically overlap, and so we collaborated with Chu and did super resolution imaging. This is the scan over here. And basically what you see is that there's just a few points of contact between Shelob, and that's what the white represents, just a few points of contact between Shelob, shown here in green, sorry for keep switching the colors, and HP1 in fuchsia. All right, now, Shelob actually behaves like an independent condensate, that it exhibits liquid-like properties. If you look by FRAP, it's highly mobile, just like HP1, not like H2A. But I think if you look live, it's really a lot of fun, because what you can see are all these liquid-like properties. You can see nucleation, growth, and fusions going from M to G1. And what you see as you go from G2 to M is some hypercondensation and dispersal. And in fact, there's this really amazing pattern. Sorry, I won't go too deeply into this, but remember I told you that in mitosis, HP1 and nucleoli dissolve, okay? So what happens with the Shelob protein is it's surrounding the HP1 and has this web-like pattern in the euchromatin. And when HP1 dissolves, so you'll see here, all of a sudden, all the Shelob rounds up. And so anyway, that's just liquid-like behavior. It then dissolves a little later in prophase. So we think that Shelob is an independent liquid-like condensate. And so it's a very strange protein. It fills the interchromatin spaces in the euchromatin, excludes chromatin, behaves like a liquid-like condensate, and it infrequently contacts the PCH condensate. And yet, it's required for pericentromeric heterochromatin organization and function. So this is not behaving like your typical biochemically defined interacting protein. What we think is happening, and this is based on doing analyses of HP1 mobility using different types of methods, FCS, FRAP, hexanediol treatment. The bottom line is that if you knock down Shelob, then you see a significant increase in HP1 mobility in both the euchromatin and the heterochromatin. And if you overexpress it, you see a significant decrease in mobility of HP1. We've done flip experiments. You also can show that in the absence of Shelob, you get more HP1 diffusion from inside to outside, the HP1 domain. So this suggests, we think that the answer is that Shelob regulates HP1 mobility and diffusion, and that's how it affects the organization and function of the PCH. So again, these are ideas, but basically that maybe Shelob behaves as a shield that helps recycle HP1 into the domain, or it somehow recruits a stabilizing factor. But the bottom line is, it's not behaving like a typical binding partner where you would say, oh, it's impacting it simply because it's binding it throughout. And in this case, it's acting in a sense at a distance, but there is this connection. All right. So just to end, how does this bring us back to the question about how layering occurs, how different condensates interact? And we have not modeled this yet. We're working with Sam now to try to bring this into the same framework, coarse grain framework that we did for the nucleolus. But the idea is very similar. The idea here is that Shelob has self-association and HP1 has self-association, but that the ability to interact, and these are weak interactions, but I'm saying this is stronger than the ability for these molecules to interact with each other. Okay, so this is a weaker interaction. And so the idea is that stronger homotypic interactions, that is homotypic with respect to each condensate, so HP1, HP1, Shelob, is what drives the formation of separate condensates as they emerge from this diffuse pattern into this separated pattern, so demixing. But that it's the weaker heterotypic interaction that is between Shelob and HP1 that drives the actual layering, the association of those condensates together. So very similar to, think of this as Pichun and HP1. In this case, now it's HP1 and Shelob on the outside, but it's the same principle. And so, in fact, we have shown, I'm not going to go into it, we have shown that the PXVXL on Shelob is in fact a weaker PXVXL compared to other HP1 interacting proteins. And that's, we suspect, based on structure, that's also true for Pichun. And so this predicts that altering the concentrations or affinities will disrupt the condensates and the layering. We've started to do that. If you overexpress Shelob, so you now increase this interaction by increasing dosage, you actually get mixing of HP1 and Shelob. And we can show that that's dependent on this PXVXL. If you mutate the central V, then you in fact no longer get mixing even with overexpression. And then conversely, we've started to play with, okay, if we decrease the strength of the self or homotypic interaction, in this case by decreasing HP1 binding to K9 methyl, then in fact you can again now see mixing for enrichments of Shelob out in these euchromatic regions. So bottom line is, we think that if we now bring these together, that this layering Shelob, HP1, the nucleolus is mediated by affinity hierarchies, specifically amphiphilic proteins with weak heterotypic affinity, contrasting that with stronger stronger homotypic affinities. And the important point here is you don't need to have a protein that just sits at the surface of one condensate to allow it to be interacting with the other. These are proteins that are actually distributed through hundreds of microns away, but it's just that at the surface, because they're held together in their own condensate, they can at the surface interact weakly, and that's what we think is keeping them together. And again, I think we need to think about, when you have these types of interactions, the implications for genetic studies, et cetera, et cetera. All right, with that, thank you for your attention. Thanks to quote team, well, it is really a team now, but it's mostly in my lab, it's just Varsha. Really talented postdoc, but we've had a really great collaboration with Sam and Omar and Gaurav. And then the Shelob work that I talked about was predominantly Sarapin. And thank you. Thank you to our funders as well. And sorry to the rest of the lab. Yeah.
Right, we still have wonderful talks today, but I have to say that what we witnessed already this morning, it feels like future of medicine. We're literally witnessing the future of medicine and biomolecular sciences. We'll take 5-minute break right now. If anybody wants to get some water, tea, get caffeinated, feel free to do so. We'll be back in 5 minutes.
How are you?
Hi, can you hear me?
That's what this. Yes. Oh, I didn't know that.
I've never seen. And I know it is. I completely agree. But no. I like exactly. I was thinking about his hierarchical autonomy aspect. That sounds almost like timing. It would be crazy if someone proposed that as an explicit timing. So they would I would argue. Yeah, but you know, I mean, I think your first point does bring down to a finding for the public.
Yeah.
Like,
yes.
Yeah.
That's what I'm
Yeah. Yeah. I
Interesting. Transcribe.
But yeah.
Reorder.
Yeah, I recognize it by
Really polish.
Okay.
Yes.
Yeah. But it's like you should never pop up. Yep. There you go. Yeah, I did a bit of back in. Yeah, I know. And what really interesting to figure out a label specifically side. Oh. Just talking about okay, you know, the predecessor to never mind.
Hello.
Really? I think myself is relatively terrible about names. No way. Yeah. This is all dating back to 2018 when I had people can't. And I was just like, oh God. I need to myself figure out all the literature and every citation. Yeah. God. I almost caught myself on the stage gave her little like thanks to the lab with all the ego creators. She had a paint packet for orange bow. And I was like, oh damn, I remember his favorites. Yeah. But he was it's hard because his last name is Clodagh, but a lot of like search properly on and so if you look for papers, Google just said, nah man, like go again. Yeah. But no, they were the people that go like in situ single cells and they almost visualize individual mechanisms and trio TMT, but like they invented like no, yeah, they're yeah. No, they yeah. Yep. And they, I mean, like 5 years ago, there was a big question. Can we find one specific images? Yeah, it'd be cool if you did, and they do that right now on public. They like engineered and like exactly cells. Iron, It's like a cylinder that accumulate ions and like acts as like a store for them.
Like,
Yeah. Yeah, and so because it has so much iron there.
Oh, yeah.
You can literally see it as a dot in cryo-EM because iron. And so they were people like repeated yeah. And then, of course, by the end of the Chinese, it's like animals, computational collaborators who were interested in working with EMG and modeling, biomolecular interactions in these two hierarchical protein structure.
Yeah, literally. I know, that's why I look at the pictures like I love like no, it did. Yeah, I mean, other people like otherwise like
So by definition, you like some of But I do like this. Like, yes.
Hello, can you hear me?
Yes. I can hear you. So I think we're all set. Yeah. Okay, we'll be starting back up.
Hello, you just want to test the audio.
Yes.
Can you hear me from the classroom?
Sonia, we can hear you. Can you hear us?
Yeah, great, wonderful. Thank you.
Okay, good. So here's what we're gonna do. So we're finishing up the break right now. In the next minute, we're gonna introduce you. You give your talk, and then there will be time for the Q&A panel, and you will be part of it virtually. Is that right?
Sounds good. Sounds
Okay.
good. Great. Thank you.
just 1 more minute.
Sounds good.
Thanks for not sitting in front of I think you were sitting here. No, so that was perfect. Definitely. Yeah, exactly. This is a common issue.
All right, welcome back. Welcome back. We have one more talk before lunch, and it's my pleasure to introduce Daniela Matei. Daniela is a physician here in Freiburg, and he is an internationally recognized leader in the field of epigenetics of chemoresistance, a friend of mine and a colleague and collaborator as well. And Daniela, thank you.
Thank you, Vadim. To continue with the futuristic team of medicine of the future, it's my pleasure to introduce Dr. Song Lee, who will speak to us from California on mechano-epigenetic engineering for cell reprogramming. Dr. Lee is a Chancellor Professor and the chair of the Bioengineering department at UCLA. He's a leader in the field of mechanotransduction, cell engineering, and regenerative medicine. And his field of research spans from engineering to medicine, a truly interdisciplinary leader in the field. Dr. Lee.
Thank you. Thank you very much for the kind introduction. I also want to thank Vadim and the Center for Physical Genomics and Engineering for organizing such a wonderful symposium. I have learned a lot from the previous two talks. And I'm sorry I cannot be there between meetings, but hope this will work out. So today I want to talk about some of recent studies on mechano-epigenetic engineering. The research in our group is focused on cell and tissue engineering. And on the one hand, we are interested in understanding how biophysical factors in the microenvironment, such as mechanical factors, electrical signals, together with biochemical signals and biological molecules to regulate cell functions. And on the other hand, we are interested in integrating what we learn together with materials engineering, drug delivery, and stem cell engineering, immunoengineering, and use that knowledge for in vivo tissue regeneration and disease therapy. So today I want to just focus on our recent work in vitro to engineer cells by epigenetic modulation. I was trained as a mechanical engineer, and we all know that in the body, at different levels, from systems to organ, tissue, cell, and molecular level, mechanical forces exist everywhere. And our focus is at a cellular level. Is for tissue engineering. If you can engineer the cell, which is the building block of tissue and organ, then we can reconstruct the tissue and organ together with extracellular matrix in scaffold, etc. On the other hand, we want to understand how at a molecular level, the mechanical factors can impact the cells. So half of our lab is focused on mechanobiology and mechanotransduction. In the past 20, 30 years, and a lot of work in the field, and this has been widely studied. And we all know that in the microenvironment around the cells, there are many physical factors can impact cell functions. For example, in the cardiovascular system, we know there's a fluid shear stress due to the blood flow and pressure changes, etc. And also cell adhesion substrate. There are many components here. In the tissue, you may have a dynamic mechanical stretching, and also the micro nano topography of the matrix can affect the cytoskeleton organization through the cell adhesion, and then affect the nucleus in the chromatin organization. In the past 10 years or so, the stiffness or viscoelasticity of substrate has been studied as well, and a lot of interesting work in this area. In addition, even the distribution of the ligands, in the chemical ligands, could affect the cell functions. And the cells need to integrate all of this information and make decisions to migrate, differentiate, proliferate, etc. So today I want to focus on a couple of things. One is the cell adhesion related mechanotransduction, also in the cells. So in the past, most of this paradigm is on how these signals can be transmitted from the cell surface to the cell nucleus. And we know that many signaling molecules on the cell surface, ion channels, adhesion receptors, they all can serve as mechanosensors. And then through signal transduction, very similar to what could be activated by growth factors and ligand receptor binding, and these signals can be transmitted into the cell and then activate transcriptional factors and regulate gene expression. In addition, there could be a direct mechanical coupling through cytoskeleton that can have a physical impact on the signal transduction as well. So many of those reports in the literature focus on the transient effect of mechanical signals. For example, the ion channels or integrin activity of MAP kinase, etc. And many of those are kind of adaptive responses. It could be activated and go back to basal level. And that represent the quick responses in probably under physiological, pathological conditions that cells can sense and adapt to the environment. So what I'm interested in is the cell memory change, which is really the epigenetic modulation. And this is very important when the cells have the phenotypic changes during the stem cell differentiation and cell reprogramming. So I think this doesn't need too much introduction because everyone knows about this. I just want to make 2 points. So here you can see the heterochromatin and euchromatin structure in the nucleus. In a couple of things. One is we know the regulation of all of these modification that can control the opening and closing of the chromatin structure. You can see a bunch of enzymatic regulation that can control the histone modification or DNA modification. But we yet do not know this operating system. It's just like a computer. We know a lot of commands, specific commands that can induce certain changes. But really the principles behind this, just like Newton's law in engineering, to guide the whole system, how this works, is not clear. The other thing I want to point out is from engineering perspective, we always think, how the mechanical or physical perturbation of this could induce any changes. Right? From previous talk, you already see these some of these very, very good examples, phase separation, liquid gel transition, etc. And we are very interested in how this could be induced by extracellular factors. So besides all these enzymatic modification of a chromatin structure, and how can the mechanical factors affect the intranucleus chromatin organization? So here it shows the schematic from the reviewer. Then that shows how this could happen, and I use that to illustrate our hypothesis. So cells, they attach to each other through cell-cell junctions or attached to extracellular matrix through integrin binding to extracellular matrix. And all of the signal mechanically can be transduced and coupled to nucleus through nuclear laminar. And one thing interesting, as you have seen in previous talks in many papers, that the heterochromatin, which is unused chromatin that in generally organized in pack around the nuclear periphery. So here actually provides an opportunity for us to test how the mechanical impact from extracellular space could affect chromatin organization. So one thing possible is that when you have a cytoskeleton reorganization, that can induce some changes in the nuclear laminar, and then could have a further impact on chromatin organization. For example, the chromatin is associated with nuclear laminar through this laminar-associated domain, etc. So, and there are also a lot of enzymes distributed around the nuclear periphery, which is kind of strategic position to regulate, for example, DNA methylation, etc. So that provides a basis that the mechanical forces could have some direct impact on chromatin yet to be verified. And the other thing is at the cellular level. So we are interested in stem cell differentiation. From this Wendington landscape, you can see if we use this metaphor that the cell differentiation can be described as a ball rolling down these hills and valleys. And you can see the two things important. One is when the cells fully differentiated, you reach a stable state. We can use physics term to describe this. In between, during this process, all of this epigenetic state at each point is unstable. A small perturbation could affect the cell fate. So physically, we could think that during cell differentiation, probably there's a less barrier, epigenetic barrier in this case, to manipulate cell differentiation, and where versus this stable state, then you have much big barrier. So if you are interested in how physical factors could modulate the stem cell differentiation or cell reprogramming, so there are a couple of strategies. For example, we could manipulate those physical factors during cell differentiation or reprogramming process. And yet the most challenging part is to overcome this barrier. For example, directly convert fibroblasts into cardiomyocytes, neurons, and other cell types, or all the way back to a pluripotent state. So here I want to show you a couple examples. We're trying to do is addressing this question whether biophysical factors can work together with transcriptional factors and biochemical factors to help and facilitate these cell phenotype changes to overcome this epigenetic barrier. So here just shows in general how the cell phenotype could be manipulated, the natural and spontaneous differentiation from pluripotent cells, and go through all these pathways to specific cell type. And now we know from Yamanaka and many others' work that fibroblasts, this could be reversed and be reprogrammed to pluripotent cells using Yamanaka factors or various combination, even microRNA and chemical cocktails. And yet there's also a possibility to convert the fibroblasts into other cells, for example, neurons, by direct conversion. So I want to show you briefly one or two slides on this part. This is a previous published work, and we'll focus on this model, which was developed by Wernick's group at Stanford to directly convert fibroblasts into neuron. So the reason we use this model is that compared to stem cell differentiation, when we do a cell reprogramming experiment, in this process in general happens relatively quickly, especially in the first few days. And those epigenetic changes could determine the long-term fate of these cells, and then could be easily captured in an experimental system. So here I just want to show you a slide of our previous work 10 years ago, in when we try to manipulate the cell morphology, especially the nuclear morphology. So what we did was to culture the cells on micro pattern surface. In this case, parallel microgrooves in the cells show very elongated shape. And when we manipulate the microgroove size to about 10 micron wide in 10 micron spacing, in this will impact nuclear alignment and nuclear elongation as well. And at that point, we could see there's a global change of histone modifications. And here we perform single cell study. In this case, we use micro contact printing to generate individual matrix islands. And then we can control the shape of these islands. And one, for example, is a circular shape. The other one is elongated shape. And these two kind of shapes, they have the exact same area, but different shape index. So here just show you one example. If you stain for histone 3 acetylation, H3K4 methylation, trimethylation marks, and these are all the marks for euchromatins. And we can see this increase globally, and also specifically at certain sites of promoter of pluripotent genes. We did ARCA4, SOX2, and NANOC as example. At the promoter of ARCA4, we didn't see much change. With the cheap PCR, we could see indeed the ACH3 increase dramatically at promoter of SOX2, and also the slight increase of trimethylation of H3K4, and a NANOC promoter at different of these histone marks increase. One is ACH3, and also the dimethylation of H3K4. So that actually gave us some clues that indeed the physical change of nucleus could have some impact on the histone modification, and then the potentially the accessibility changes for gene regulation. So here is a work in revision, and what we want to do is directly test if you relax the intracellular tension by the disruption of actin-myosin interaction. Right? So if the hypothesis is right, the connection between the cell adhesion to nucleus is through actin cytoskeleton. If you disrupt this, what will happen? And I will just show you a few slides on some important data. For this case, we use the ABM, the 3 transcription factors identified by Wernicke's group. And what we found is that during the reprogramming process, ASCL1 specifically, the increased cell adhesion, cytoskeleton organization, actually that could cause form a barrier for reprogramming. Efficiency is low. If you do embryonic fibroblast conversion, efficiency is reasonable, but if you use adult skin fibroblasts, efficiency is 1 magnitude lower, down to single digit or even less than 1%. So what we did was to disrupt the actin-myosin interaction by using blebstatin, which is commonly used chemical interference. And this shows a dose dependency on the reprogramming efficiency to convert fibroblasts into neuron. At a low dose, you have some slight enhancement. At a 10 micromolar seems to be optimal. Of course, if you go beyond that, it could be some side effects, because cytoskeleton is involved in so many different things and can affect various signaling events as well. So now the question is whether this cytoskeleton disruption directly regulate this neuronal gene expression or indirectly. So we did one experiment, and in this case, we could just test fibroblast cells without any reprogramming factors. BAM, it is 3 reprogramming factors, or with the 3 reprogramming factors. So in the absence of reprogramming factor, you just take fibroblasts and treat them with blebstatin. And for mesenchymal gene, I didn't show you here, indeed, when you disrupt cytoskeleton, there's a decrease of expression, which helps because if you want to convert mesenchymal cell types into neuron, which is epithelial type, there's this mesenchymal to epithelial transition. That's helpful, but that's through a different mechanism because it's known that some transcriptional factors related to actin polymerization, their translocation into nucleus, such as MRTF1, etc., could regulate that process. But for neuronal gene expression, these neuronal genes are turned off in the heterochromatin in fibroblasts. They are off. So when we add a blab, and you can see here the first 2 bars, and almost nothing for these genes. So disruption of cytoskeleton alone does not induce this gene at all. Yet when you have the reprogramming factors introduced into the cell with viral constructs, and then you add this blebstatin, you can see this enhancement compared to the control with DMS cell. So this shows that there's some kind of a synergistic effect. The cytoskeleton is doing something, and potentially through epigenetic modulation. Then we examine a few histone marks. We also examine the DNA methylation as well. And by the way, when you disrupt the intracellular tension, the DNA methylation globally also decreases. I didn't include that data here. I just want to show you a few. ACH3, H3K4 trimethylation, H3K4 methylation. And for all of these, when you treated cells with blebstatin, they all increase. This is globally. And how does this happen? It's possible, as I said, it could be a mechanical effect. It could also be chemical enzymatic effects through all of these enzyme HAT activity and etc. So in this case, when the cells in culture, when you treat cells with any chemicals, actually, I think it's there's a combination of mechanical and chemical effect, because there's a chronic effect. There'll be time for the signaling events to happen through hours or even days of reactions. So indeed, when we examine all of these enzymes, HAT activity increase dramatically. And on the other hand, HDAC activity decrease. These are all global nuclear extraction assays. And in addition, the HMT activity for H3K4 increase as well. And then the HDM activity, which is inhibitor of the methylation or decrease. So this all correlates so well. And whenever we observe these global changes and examine this enzyme, it seems the enzyme all change in the same direction. And now we want to see whether specifically if the hypothesis is right, the cytoskeleton disruption, it could enhance the accessibility of some of these sites in heterochromatin, for example, neuronal gene, the promoter of neuronal gene. So we did an ATAC-seq. We generated a genome-wide map of accessibility and look into a few examples. Here, I just want to show you a few sites. For example, beta-tubulin, the neuronal marker, ASCL1 and MYT1L. And you can see the promoter or enhancer of these genes. There's a big increase at some sites, and you can see the difference. So that suggests that it is possible when you disrupt the intracellular tension, you increase the physical accessibility at certain sites, and that together with the reprogramming factors that allow them to bind to these sites in the heterochromatin to enhance cell reprogramming. Indeed, we also analyzed the ASCL1 targeting site. ASCL1 is a pioneer factor for reprogramming, could help open up the heterochromatin structure. And we found many sites are also open after you treat cells with blebstatin. So that's one part of work. We're trying to see how this intracellular tension could affect nucleus. The other thing we want to do is, in a literature, there are already some reports showing various cultural conditions, the stiffness or mechanical loading could affect the epigenetic changes. And yet there are some conflicting reports on how this could happen. Sometimes people showing increase or decrease of certain modifications. So I think in general, these microenvironmental factors, biophysical factors, is a context-dependent regulation. So when you have cell adhesion or even different cell types, the matrix adhesion in cytoskeleton, cell spreading, all of these could have impact. So we want to do is now let's remove all of these systems, remove the cytoskeleton, everything, and just bring cells in suspension. And hopefully we can directly perturb the nucleus in a way to induce a nuclear deformation and see what happens. So hopefully that's a more clean system physically to perturb the nucleus. So in the literature, people have used various techniques to achieve that. Micropipette aspiration, you have a negative pressure here to suck in a part of a cells or nucleus. And by the way, all of these technologies have been used for single cell or even single nucleus manipulation. In atomic force microscopy, you apply a compression, and then you can one is to measure the mechanical property changes, and two is also induce some changes physically in the cell nucleus or optical tweezers at a local. This is a more kind of a local manipulation. So all of this will give us some really detailed information in cells or nucleus at the single cell level or single nucleus level. Yet it's a low throughput. For us, if we want to figure it out, for cell differentiation reprogramming, and also some molecular analysis, we need a large number of cells. So we decided to go for the very last technology platform is shown here, which is microfluidic system. So this system actually, there's also some flexibility to manipulate. For example, you can manipulate the sizes of channels to control the magnitude of compression or deformation of nucleus. You could also have these cyclic changes of the shape, so you can apply either constriction or relaxation to cell nucleus cells in a cyclic way. So we use this system to perform the study to test directly how nuclear deformation could affect the chromatin organization in cell reprogramming. So here it shows the experimental procedure. We first introduced all these reprogramming factors, BAM, 3 factors, and this is a DOX-inducible system. So we activate these genes, and then transcription factor can be expressed. We have tested this actually time course between 6 to 20 hours, and that's an optimal time to induce cell nuclear deformation. So this nuclear deformation is very fast, and this is very different from what we did in the cell culture, which takes hours or days to see these gradual changes in cell adaptation. Instead, here is a microsecond. I think it's even shorter. I need to just go back to. I think it's a microsecond deformation of cells. A millisecond, I think, millisecond, sorry, just make sure. And we're also trying to make a high throughput device to even have a shorter nuclear deformation. In this case, the idea is to make something similar to flow cytometer, and we can have a microfluidic system applying this quick deformation of cells within minutes. You can process millions of cells and then use that for cell engineering. And then we culture them for various lengths of time to look at the cell reprogramming, maturation, etc. And there's one thing interesting and also important too for this system. When we apply the physical deformation to cells, we need to be careful. Otherwise, it could damage the cells as well. So this is a cell size dependent when you use this microfluidic system. And here it shows how we characterize the fibroblasts, even for mouse, adult mouse fibroblasts. It's heterogeneous. You can see this Gaussian distribution, cell diameter, but the most important, we care about the nuclear diameter. And here is about 10 to 11 microns in average, but there are some cells bigger and some smaller. And if you use different sizes of the channel, and by the way, for the convenience of microfabrication, we make all of this channel height consistent, which is about 12, 13 micron, and we only vary the width of these channels. So you can see here 3, 5, 7, 200, etc. We did a various characterization just to make sure we don't kill the cells. The cell viability is very important. For example, here you can see this is a laminin AC staining with H2B GLP labeling in the cell as well. If you have a small channels, you can even cause this blabbing and disruption of nuclear membrane, etc. And what we found is 7 micron seems to be appropriate for this cell type. So which is smaller than the average. That means you do have a deformation of nucleus, and then yet you don't induce significant cell death. And here, a wide channel we use as control. We just let cells flow through a big channel without a compression, but they are subjected to the same fluidic exposure. So one thing we noticed that when you subject the cells to this mechanical compression, and you definitely deform the nucleus, and you can wait after experiments. You can wait for hours. You can see the cells to the surface, and the cell will take hours to spread out and get back to normal stage. And we measure the stiffness of the nucleus after 3 hours, 16, etc. And in this case, we use AFM to directly compress this cell topic. This top right at the top of nucleus site. If you do this measurement near the edge of this, you probably will measure the cytoplasm mechanical property. And if you put on top of this, it has been shown in the literature, the major contribution of this stiffness is from the cell nucleus. And you can see there's a definitely decrease of nucleus stiffness right after mechanical squeezing, and this gradually recover. So I think this is consistent as what we have seen in the previous talks. For example, it could be this gel to liquid transition, and you disrupt certain structure and probably change this DNA organization. And then we examine the reprogramming efficiency. And here just shows the time course. If you look at the ASCL1 gene expression, indeed within hours, if you squeeze the cell, there's a huge increase. Of course, this is a total ASCL1 expression, including those forced expression of reprogramming factor. And then we use the ASCL1 promoter-driven GLP to directly examine the endogenous gene expression. Because what we want to achieve to reprogram the cell is to activate the endogenous neuronal genes. And we indeed show it's a significant increase as well. The other question we wanted to address is whether this mechanical deformation of nucleus make this reprogramming process faster or earlier. And we use these nano probes developed by Pak Wang's group at Penn State. In this case, you could detect early mRNA expression of the gene, neuronal gene. In this case, we use TUB3 as a marker. And you can see indeed with mechanical stimulation, and you induce this neuronal gene expression endogenous earlier. So the reprogramming process actually happens very quickly. And we found that actually the first day or two are the most important. And afterwards is reorganization and remodeling. So what happened to the epigenetic marks? We again look into these all of these histone modification in the supplemental data. We listed, I think, over 10 different kind of histone mark stainings and DNA methylation as well. Here, what we found is one major changes in this mechanical regulation is H3K9ME3, which is the heterochromatin around the nuclear periphery. And if you look into here, control, and you can see still a lot of these clusters of DNA chromatin here, and with the squeezing, you see that they are dispersed and form this really diffuse pattern and gradually recover within 24 hours. So this probably is also the indication that some phase transition and other changes, and we yet need to look into. And also, what we think is that this probably is the time window if you want to reprogram cells, and this very similar to embryonic cells or other cells, which is kind of a primitive or a stem cell state that the chromatin yet not to form has not formed so many this kind of a closed structure, heterochromatin structure yet. And this is also changing. We quantify this process. and now the question is, people know that the enzymatic effect on the heterochromatin or just general chromatin mark changes, histone mark changes will take time. How fast this physical induced heterochromatin mark changes, specifically H3K9 in this case, will happen. How fast will happen? So we use this FRAT probe developed by Peter Wang's group at the UCSD. In this case, it detects the binding of H3K9 methylation in HP1. We put this probe into the cell, and here shows a one cell, a time course, because this takes really millisecond for cells to go through. It's too fast to observe these changes. So we slow down this process by adjusting the pressure in the microfluidic system. So we lower the pressure and allow the cells to slowly go through, but this just takes a few minutes. You can see here, red color indicates higher H3K9 trimethylation level, and green means lower. And when this cell passes through, and this shows the nucleus shape change, and the signal decrease within minutes. It's very fast compared to traditional, whatever people have reported for chemical modification. The other thing we look into is DNA methylation. So similar to what we observed for the H3K9 trimethylation, there's a decrease of this clustering of DNA methylation that indicates that there's a decrease of this closed structure, and this is also transient. So what happened in a cell? Is that everywhere increase or decrease? So we did some experiments. These are unpublished. We did IC in ataxic in sequencing to trying to figure out what happened genome-wide globally. So it's interesting if you look into this heat map, ataxic data. We showed a duplicate for two samples. So when you mechanically deform the nucleus, about 60–70% of the sites become more open, but actually there's about 1/3 of the sites become more closed. So it's definitely there's some physical and spatial effects, and we also found some of chromatins are overrepresented. In this case, there are more changes. So if you're looking high-C data, we need to analyze those. And here just shows a couple of examples that a specific promoter or enhancer of ASCL1 in tube 3. It shows the peak changes and accessibility changes. In general, several neurogenes promoters all shows the increase of accessibility. And the other question we address is, how can we compare this biophysical effect with biochemical effects? So that well known, many of these chemical inhibitors that can modulate, for example, here, the BIX is H3K9 specific methyltransferase inhibitor, and then the Cetobin is the DNA methyltransferase inhibitor. So we add them alone or in combination and compare that with physical squeezing. And here showing that if you have squeezing, you have increase of reprogramming efficiency, and together there's some additive effect, but not too much. And for the DNA methyltransferase inhibitor, it also shows some enhancement, and then together with mechanical squeezing, this is additive. And we also try to just have a chemical combination, because now these two changes, H3K9 trimethylation and DNA methylation, if you suppress both with chemicals, actually they reach similar level as physical. So physical squeezing of nucleus actually causes more changes, and maybe that's something we haven't examined yet. And now the question is about mechanotransduction. When you deform the nucleus, in this case, all cytoskeleton have been disrupted in suspension. And what happened? So one thing we look into is the nuclear lamina, which is the structural support of nucleus. And if the hypothesis is right, then always heterochromatin affiliated with nuclear lamina could be perturbed. And here we show that if you squeeze the cell, you see this wrinkling. And definitely you make the nucleus softer as well. And then this gradually recover. And also at the beginning, if you quantify this lamin AC at the nuclear periphery, there's a slight decrease in represents a partial disruption. So then we directly examine the interactions between the nuclear laminar and chromatin by using this M6A tracer that to detect the LAD interaction with lamin B1. Here shows lamin B1 in red, and M6A tracer in green. And after mechanical squeezing, you can see some partially at the periphery of the nucleus. You decrease the direct colocalization of heterochromatin or chromatin structure with nuclear lamina. So if this nuclear laminar perturbation really plays a role in this mechanotransduction, then we directly disrupt the structure. It should work. Indeed, actually, in embryonic stem cells, people have reported that you have much less nuclear lamin compared to fully differentiated cells. In here, we use siRNA for lamin A, and you can see when you disrupt nuclear lamin A, you also see a disperse of this DNA methylation over here, very similar to mechanical squeezing. So we quantify this H3K9 trimethylation and also DNA methylation, and we found that indeed there's decrease with lamin knockdown. And the lamin knockdown also increases the reprogramming efficiency when you're trying to convert fibroblasts into neuron. And this is consistent as our hypothesis that when you disrupt the lamin matrix, you probably will increase accessibility in chromatin. So the last thing we trying to figure out is whether this is a general phenomenon. Is that cell type dependent? And we try other cell types. For example, a challenge is to convert fibroblasts, which is easily obtained from circulating blood, into neuron. So in this case, we deform macrophages in a microfluidic channel, and we perform the same experiment as in fibroblast conversion. And we show that in this case, the mechanical squeezing also increase the reprogramming efficiency. And at the same time, when you convert fibroblasts into iPSC, you see much more colonies if you subject the cells to mechanical squeezing here. And we are trying to do is make high throughput microfluidic device. This is just one design with this reading configuration, and also some other designs we're trying to do is a smallest space area. We can put as many channels as possible to process many cells in parallel. So in conclusion, what we think is that the intracellular tension, the actin cytoskeleton, which is important to connect cell adhesion to nuclear laminar, definitely plays a role. If you relax this tension, you probably make some free-floating nuclear situation, allow easier chromatin reorganization. And if you directly deform cell nucleus, as we shown in this microfluidic system, that actually has some impact because this directly affect the heterochromatin at the nuclear periphery. And we believe this finding could lead to certain applications. In this case, what we are trying to do is generate a Mechano-Epigenetic map following this nuclear deformation. And we found this is consistent. And if we identify certain sites with increased accessibility, we can use the CRISPR to design specific guide RNA in target those sites and demonstrate it is true when you have mechanical perturbation. And locally, you can also engineer gene activation or deactivation with CRISPR-mediated gene editing. And we are trying to make a high throughput device, and hopefully could have some application for cell engineering. And with that, I want to thank the funding source, the collaborators I mentioned earlier, and also the cytoskeleton work was led by Jennifer Sato, a postdoc in the lab, in the mechanical squeezing project was led by Yang Song, another postdoc in the lab. Thank you very much.
Thank you, Son, so much. And by the way, Son was a little too shy. The reason he couldn't make it today because he's receiving a major award, and it was either us or the award. And but he did ask me, "What should I do?" And I said, "No, get the award." So we're happy to have you. Thank you so much. Now we have time for the Q&A session. I would like to invite our esteemed speakers right here. Let's we have a little table here. Let's get the chairs. And Son will just project your face, I guess, on so people can see you. All right, second. All right, I think everybody can see you now. Like I said, ask your questions. Every question is welcome. I'll just, to get the conversation going, I'll ask the first question. So in your view, and that's a question to everybody, in your view, what are the major unanswered questions, million-dollar, billion-dollar questions in the field of genomics? And is there any technology that doesn't exist today that we're desperately missing to answer these questions? Actually, yeah, so let me sure you have microphone. Right, yeah, microphone.
Oh, I see. Okay.
Whoever wants to start, in no particular order.
Okay. Yeah.
Yeah.
All right. Okay. Yeah. No. So I think. Let me see, John. That's the control. I mean, I think. I mean, Gary kind of brought it up actually. And again, this speaks to kind of your work as well, Vidyam, in terms of integrating dynamics actually in live cells, kind of with high resolution structure and function, right? Is that, you know, we have these kind of genomics basically where it's sort of, you know, sort of global population views of everything, but none of them are kind of connected. So we can measure individual things sort of at a population level often, but then how do we connect that back to individual cell behaviors basically and the dynamics of them, right? Where no two structures might be the same, right? Which is a very different way of thinking about structure, right? As a biochemist, I want to impose the three-dimensional structure on top of the three-dimensional structure. But I think the lesson might be that they're statistically repeating sort of in a dynamic way where there might be on average certain contacts that give you sort of this kind of critical phenomenon, et cetera, mediated by different factors. And that gives you robustness as well as variety, right? And so I think what we really need is sort of, and again, I think Vidyam sort of can speak to this, is in terms of building multi-scale maps sort of cells and cell phases, is a single technology that could actually sort of ideally enable us to measure, you know, transient dynamic interactions and actually sort of memorialize them in a cell, right? So we could find them after by mass spec in an ideal world by genomics, right? So we could see them and then even begin to sort of combine that with actually then kind of visualizing the structure. That would really give us, I think, kind of structure to function, to sort of phenotype. And, you know, I think this is the power, I guess, of genetics, and I'll hand it over to you, is that I think the issue with imaging, to be fair, right? Again, the weakness of imaging and the power of genomics, they're both sort of opposite of each other, is that the issue with a lot of imaging experiments is we kind of have 2 or 3 colors and we only label what we know. We don't see what we don't know. So they kind of become self-fulfilling prophecies. So we, you know, so how do we sort of look at, you know, can we develop things that detect phase detectors and begin to proximity label them, right? And actually that same technology in genomics and kind of imaging, right? I think that's a huge gap, and the same technology that allows us to integrate genomics, right? Single cell population. That's kind of my ideal dream, right? This kind of multi-scale.
No, it's hard to add to that because I think that's exactly, I agree completely that, you know, and I think that that's the big challenge is in vivo, it's very difficult to deconvolve the multitude of interactions that are occurring in time and space. And you know, really, you said it beautifully. I mean, the technology is not there. There's absolutely no question that we can't do that type of thing yet. I think we're making advances. Two things that I would add, and I'll preface this by saying I don't do math. Unlike many of you, I'm not a physicist. My older sister is a solar physicist, but none of that got, you know, none of that got transmitted to me. But I've over certainly the last 10 years have come to really appreciate the value of integrating physics and physics theory into the biology. This is not easy from both sides. I mean, some people like John and Andy can do both. But you know, it involves finding the right collaborators. I'm very lucky, Sam Safran, Daniel Joes, Frank Ulicker, people who can speak to dumb biologists like me, and you know, get the conceptual component across without necessarily having the math. So, and that, you know, it is very challenging, but I think we need the technology, we need the theory, and we need to iterate, especially with live systems, in order to gain a better understanding. The other part of this is, and this is going to sound like the old curmudgeon that I am. Yes, I'm sorry. But I think we need a real fundamental change in perspective. And what I mean by this is in the nuclear organization and chromatin field, there is still, in my view, an overemphasis on, you know, high throughput, large data set types of approaches, which are extremely useful. Do not get me wrong. They're extremely useful, but they become an end to themselves. And in a way that is not as informative about the basic biology as it should be. And so if you do the pure computational genomic analyses, ChIP-C, cut and tag, Hi-C, et cetera, without doing the imaging, without doing the physics, you're missing a huge boat. And the problem, as I see it, is that it's very attractive to generate tons of data. And it gets you published in wonderful journals, but it's not telling you, in my view, what you need to understand biology. That's why.
Well, thank you. So I think what you're saying is that we need something like, you know, Maxwell equation for the electricity, right? We're not generating data on gazillion electrons. We just know how things work, right? Yeah. And get to the basis of it.
Yeah, I mean, if you want to, there's incredible use to those approaches, absolutely, and we learn a lot. But I think we can't view that as the end. The end is understanding the biology, and that requires much more integration. I think that's one of the beautiful things about your work is the integration of not only multiple scales but multiple approaches. The more orthogonal approaches we take, the more we can learn.
And Sloan, I'm sorry.
Sorry, I guess just one other thing I would add is that, you know, I do think as well, right, you know, modeling, again, you know, at the end of the day, it comes down, I thought that was beautiful in terms of predicting sort of, you know, what these molecules might be like, right? What are the scaling laws, right? And so one challenge I would say to you is that, you know, again, design is the ultimate test of understanding. You're an engineering school, right? You know, evolution is sort of blind, right? And you can discover kind of principles, but seriously, right, there are actually certain principles now that are emerging, right, in terms of amphiphilic sorts of fetuses, you know, is it sort of small repeats? Is it basically oligomerization, right? And so, you know, what would be the device that you would build to change the genome and to create entirely new cell fates, right? That's sort of, I think, the ultimate challenge to understanding. And that's where we could bring it. Imagine if we could design completely new things, right? And I actually think that some of those principles are there now. And so it needs a combination of synthetic biology. What has nature made? But it hasn't explored endless possibilities, right? What is the device that you would make? And can we basically sort of use them to integrate into these systems and reimagine them? Because if you can build it, you understand it. And so I think that there's also a need sort of for coming kind of from top down and bottom up, and they have great utility as well for medicine.
Very good, very interesting. I just, I see the question, just, I think, Son, you wanted to add a few words, right, as well?
Oh, yeah, I want to echo what Dr. O'Shea and Dr. Karpen said. I agree that the integration of the molecular level, high level, high resolution imaging together with genomic analysis will be very helpful. And in addition, I would say that I agree that the really interdisciplinary approaches to address this will be necessary, because I think sometimes you need a hypothesis to test, in an idea out of the box could give more insight and give some direction for exploration.
Oh, very interesting. Thank you. So let's just take questions from the audience.
Yeah, so I had a follow-up question, which is kind of related to how do you build up an understanding of these sort of multivalent interactions that there's all these different players, and they're all I'm kind of struck by, on the one hand, there's a philosophical approach, like take as an example, like Gita Narlikar did these very nice kind of binding assay studies of mononucleosome, dinucleosome, throw in, I guess this was SWE6, but like HP1 analogs, and then you get numbers, right? But of course that gets exhaustively complicated if you're talking about all these possible compensatory effects, and you had a really nice example of different phenotypes that emerge by taking out one protein player, and then it totally shifts the organization. And I guess from your viewpoint, is it how do you approach that problem? I mean, is it a matter of doing the in vivo knockdowns and seeing different phenotypes and then saying, oh, what must be the sort of hierarchy of interactions? Or do you think that there's a lot of value in that bottom up sort of approach where you're literally trying to take each individual player and figure out what those interactions would be? Is that a clear enough question?
Oh, yeah. I mean, it's
Great question. Thank you.
I'll start. I mean, it's essentially what we were talking about before. I think, each of these approaches has value and provides information. So in the case of Gita's beautiful work on SWI6, the biochemistry, at least in my view, the biochemistry in vitro, as for the sort of coarse grain modeling and other types of modeling based on the basic physics, tells you what's possible in vivo, but doesn't actually tell you that it's happening in vivo. And so again, back to iterating between those two components and others, and what you see in vivo, and the genetics and the genomics, I think is the way to go. But it's got its limits. I mean, one of the points I was making is you get rid of this factor, or you get rid of even, there's a lot of work now on, I get rid of this disordered domain in a protein, and I see, a phenotype, I get rid of that disordered domain, and in vitro, I see that alters phase separation. Therefore, phase separation regulates this process in vivo. No, we can't make, I mean, it's possible, but we can't really make that leap because we don't know what else is happening. And, but I think our best approach is to be combining all of this. In my own lab, we're trying to do the in vitro chromatin assembly a la Mike Rosen, combine it with HP1, and answer basic questions about energetics and kinetics at that level, and then integrate that with the theory and use that to inform us about the experiments we should do in vivo. But so I really don't have a good answer. I'll just add one other thing, which is that what we have found to be useful is gain of function mutations rather than solely loss of function mutations. So the problem with loss of function is, I get rid of a chromodomain. I didn't go into this in detail. If we get rid of any of those interactions, these multivalent interactions for HB1, dimerization, chromodomain interaction with chromatin, interaction with binding partners, reduce the levels of binding partners, we dissolve or severely affect the condensate, right? But the problem is you can't say that let's say the phenotypic consequences of that are because you lost the condensate. Whereas when you change the material properties by increasing affinities as one version of gain of function, then you have at least another tool in your. And so, if, for example, in that case, we can make more solid HB1 condensates that now have phenotypic consequence. It's still difficult to interpret that, but it is at least another approach. So yeah, I think what you've highlighted is the big problem. And I'd love to hear other solutions, but I don't see anything other than just combining all of these tools that we have in a way that is integrative and thus more informative than each of them.
Yeah, I think basically, I think what Gary said was exactly the thing. I think I have just maybe one thing to add is that I think one thing to think about is that, proteins and nucleus are there fundamentally to regulate the DNA, right? And again, and thinking about sort of a chromatin polymer, which in itself, as part of a chromosome, doesn't really it has much more constraints on potentially its relative movements and potentially its interactions with the lamina, nuclear volume, right? Basically, osmotic potential, ions, right? I mean, all of these kind of different things, right, will change concentration, right? Basically, local concentration. But also that polymer, right, is going to be sort of much more stable. So again, what's the size of the domain, right? 300 nanometers, where things begin to operate, because I think we've witnessed kind of a lot of these high C where people are shocked that they get rid of CTCF side and nothing happens. I'm not shocked at all, right? Because it's part of one interaction, and maybe on a weak regulated gene, that could be an important interaction. But when you have stronger interactions that drive it, it's less important, right? So again, it's an integral model of many different factors that together, right, in terms of a signal response, right, I'm building a device that now we can't anticipate, sort of an asteroid or an unknown stress, right? So we build devices that can potentially respond to different signals that could be integrated to give rise fundamentally for selection. That's the only drive that we have, right? And so that's a really interesting idea about, again, critical phenomenon, right? Is that multivalent interactions are part of a critical phenomenon. And in some cases, they may be the critical phenomenon, but in other cases, high affinity transcription factor binding site is the critical thing, right? And so again, I think it's about sort of that inherent flexibility in biology, right? Because it's amazing to think you can mutate something and survive and get away with it, right? And yet, it can give variation and yet complete robustness, right? That I think is really what's interesting about valency, right? Where you don't have to have 5 every time. It doesn't have to be 13, it doesn't have to be 70. And it can be combined with other things to push something over kind of the top, right? And this is, again, where I think repeats of DNA, right, may have completely different constraints. As a polymer, they're less diffusible as well, basically, right? It's really sort of almost semi-solid if you think about it in a packed nucleus, right? It's totally different. It's not diffusing, right? The polymer is really moving as maybe microdomains. And sort of it kind of gives you sort of ideas of sort of the beginnings of kind of that kind of I think genome organization. But I think it's really going to be important to sort of integrate the protein type liquid transitions on a semi-solid, effectively long, chromosomal like array as opposed to short nucleosomal arrays, because that's a short polymer. It has a totally different property to a genome level sort of chromosome, right? And chromosome territory.
Good, thank you. And we have a question here.
A more technical question for Dr. Osha. What is your take on the fighting between the labeling and the precision of imaging? So let me be more specific. an antibody plus second antibody plus fluorescence protein adds up to 30 nanometers.
Mhm.
We want to image a histone protein. Considering the uncertainty of rotation, flexibility, stretching flexibility, the fluorescence particle is far away, can be far away.
Right.
So based on the labeling, we perform nanodomain analysis. We perform contact analysis. Are they reflecting the truth, or because everything is statistical based, it actually be okay?
Yeah. So no. So this is a great question, and again, Gary will remember the arguments between people, the fish experiments, and sort of the IC, where people thought, oh, you should do the fish experiment. I'm going, actually, I think the fish experiment is good. So to your point is that I think that fluorescence microscopy, right, and even with super resolution down to the point spread function and the rotational, I think you can get a general localization. You can build general trajectories, right? And the size away, yes, I mean, I think this is a big point, is that when you start to put together an antibody, right, it's basically like 17 nanometers basically between each FAB, and you start to build these up, you may actually end up getting exclusion of antibodies and certain things from certain domains if they're fixed, especially sort of in fixative. So again, I sound like a broken record, but this is again where I go, it's not one or the other. It's not this is better. It's that I have more confidence if two different techniques, right, like Mano-Chia, Storm, give me the same result, right? Because and sometimes each might have a strength or a weakness, but I would hope they would converge on the central biology, right? Where live imaging might sort of suggest general kind of diffusion dynamics, and maybe that's important in interpreting some antibody fixation experiments about what's excluded or what's not, right? And even then, if we think about single molecule sorts of resolution experiments, here the issue is that we build models based on very, very tiny amount of particles, and we imagine what might be there, but we don't actually know because we can only label two things, right? So I think that again, it's the integration, right, of using sort of high resolution antibodies. There you can look at post-translation modifications, which can be incredibly important. But being open to the possibility, again, that it's not that the data is wrong, but that there may be an alternative hypothesis that should be acknowledged or potentially, if possible, excluded or confirmed with another technique, right? That's how generally I would think about approaching, right? Every single technique, there is not a single one that doesn't have its own limitations. But can we bring together more than one technique that might either arrive at the same answer or a different one? Because that's really, and it's not, don't think about it as wrong or right. Be curious, because sometimes actually things that I thought were wrong were the most interesting. So to answer your question, I would say with the precision. So what's interesting with the fire nano, that's what we were doing at the end with that, is we have a 1 nanometer, it's about 12 nanometers particle, right? And you can actually build in, that's what I was building in the structure of RNA polymerase. I could see the gap between the DNA. So there we can completely build it in. And so again, if you can kind of use that kind of an approach to really get at what you're doing if you start to use tomography in these pieces, right, with fluorescence.
And to give us some time, we just take one last quick question. We'll have more opportunities to ask questions later in the day because we'll have another panel.
Thank all three of you. Those were amazing talks. My question is, a lot of us here are engineers and like physicists and biologists, but how do you communicate kind of the physics-based principles to clinicians and people who are interacting with patients, and like what kind of ways does our understanding of like physics translate up into like human biology, and how have you kind of experienced that? Loaded question. I'm sorry.
No, I think it's a really good question. I can't.
Some physicians here.
What's that?
There are some physicians in the audience.
So I can't speak to how to communicate with clinicians other than what I've learned is my younger brother, who's a MD PhD, is he's incredibly useful to have when you have to go to the hospital. But no, I think for me, all that I'll say to that is I have had a lot of experience trying to do integration. You know, I don't even do computational biology. I don't know how to do bioinformatics, but, and again, going into exploring the physics side of things, is that it really boils down. So in both of all of these cases, there's been numerous ones, it's boiled down to communication and learning how to, you know, really understand what the needs are of that other person that you're communicating with and vice versa, right? So, you know, and so basically the bottom line is you need the right kind of people who are willing to step back and say, okay, I'm a physicist, you know, I really want to focus on the math, but I am willing to spend the time to explain to you in English or whatever language you use, you know, what this means in a larger context, and vice versa. The physicists that I collaborate with don't care about the proteins, right? I mean, they do in a way, but they really don't want to know all the details that I have up here about how chromosomes, blah, blah, blah. But, you know, both of us have to be willing to step back and communicate in a way that's effective. And I think that that's actually really hard. And it's not something that everybody can do. So that's, you know, I can't really speak to what works for clinicians. I think clinicians need it, but I think it, as with everything else, it likely boils down to who it is you're talking to, and are they willing to engage at that level, which is very difficult. You have to learn each other's vocabulary. You have to, you know, spend time. I did a sabbatical with Sam back in 2017, and it's just now that we're finally getting a paper together. I mean, you know, it just takes time, and okay, that pandemic thing didn't help. But
Well, thank you so much. And again, I know there are a few more questions we have here, but we'll have another chance. We'll have another panel later in the afternoon. Thank you so much. Thank you for your wonderful talk. Thank you. One more thing, I just enjoying really listening to this beautiful talks and learning so much. But there's a lot of organization that has to happen in order for us to get together, and I'm not the one really responsible for this. So I would like to take a moment and thank people who actually who have put together this event and spend a lot of time doing that. We all know how much effort it actually takes. We have Benjamin Keane right here. Okay, great, thank you. Rosemary, somewhere. Oh, she's okay. She's organizing the next thing. All right, and we have quite a few volunteers who are most of them are trainees at the Physical Genomics program. If you just stand up, show yourself. I just want to say thank you. Thank you so much. We're all really appreciative and grateful to you. And now Ben will say a few words about the logistics of what's going to happen next.
So just very quickly, we've got an hour for lunch. So right across the street, you can go out these doors. Just to the right across the street, you'll see the glass atrium. We've got box lunches over there. There's a couple dozen wonderful posters. So please go over, grab a lunch, mingle. There's lots of smart people here. And we'll plan to start again at 2:15. So that gives us exactly an hour. And we'll be right back here. Thanks, everybody.
no, it's not. It's not. No. I just mean that. It's compound.
Oh, no, that's.
Yes, on Friday. I was going to tell you. Abby. Yeah, how am I? Yeah. Oh, well, that's nice. Actually, that's a good. It was I have to teach that. Yeah. Well, some other. I have to know. And it would be somehow it'd be great to have you out. There's a lot. Yeah. I mean, I love James. Well,
How's it going, Raymond?
let me also see Aaron. Yeah. Oh, okay. Right up here. Oh, okay, yeah, you had it. Yeah. Yes. No, it's not. Yeah. They're in the streets on this, they seem to be right upside. Right upside. No, just asking like gypsy. Truly a lot of fun. Yep, street straights. Well, let's. Yeah. Yeah. Oh, because I want to say, I assumed that maybe plus. Yeah, we looked for RNA, we didn't find her. No, I'm not saying that they're. We did see. What's what? Just this, yeah. But we do find that.
I can't do it.
And we're very interested. Also study, I want to see that comparative proteins of the normal and mutant or the mutant or minus RNA cell. Did you compare the profile
Protein, no, we have not. Yeah, that would be. Yeah. You going?
Yes. Sorry. I. Get lost.
We cannot get all of it. You can just drag it right now. Can you be dragged it around?
Sorry.
No, please go ahead. That's good. Did you get a chance to grab lunch?
Yeah, yeah.
Okay, I just came to look for you. I'm like, I didn't see you.
No, I have a collaborator. So,
Okay, okay.
do I need to load my stuff?
There's an AV guy that'll come.
Okay.
Don't worry, if he stops by, actually he should come at 2, so I'm
Sure.
gonna just hang in
Yeah,
here.
don't
You
worry.
sure you don't want lunch? I can get you a
Oh, I had
box.
a lunch.
Okay.
I had a great lunch.
Okay,
Yeah,
thank
they
you.
took
so
me
much.
out.
Okay. I was like, I don't see you. I was like, where is he?
I told Rodan
They had another meeting, so they went somewhere, so maybe he didn't get a chance to tell any of us. Thank
Thank
you.
you.
I'll let him stop by. I'm right.
Don't worry.
here.
Yeah,
Thank
thanks.
maybe I can use his laptop itself, whatever is easier.
Whatever works for them.
Whatever works for you.
I can load my
Yeah,
pocket.
if you want to just open out of there.
Yeah, sure.
I'm gonna swing by there and be back.
Okay.
Okay, fine. Thank you so much.
I'm the second one. Okay, great. Now, is it fine if I leave it up like this? I can just close it. Somebody else is
Maybe these are the best ones. No, just help our students. That here. We want to sit. Front row. Is the too close? Yes, good. Middle. We will be on P. Sorry. Now I have this is already. I see. Okay, can sit there. Yeah, yeah. Where do you want to sit? You sit over there. This here. There we go. It works. But it's naked here. It was so cool at some point. I didn't think it was. This was when I was so but I think when we were there, great. I'm getting this almost too much. It's almost too cool, but if you want to be part of something, you don't. So how about then? Holy. Put a little note. But I think to make this anyhow. I need to be. Study educators at the Biochemistry is both breast cancer related and also. Yeah, it is. Yeah. What? So use little bit. It's kind of good. Yeah.
letting us know.
And we're looking forward to working together.
Yeah, hopefully I should be able to tell you guys this week. I don't know, have you been in touch with John at all about CRISPR logging?
Not later.
Because I think he's the one who's at Brown, who might be making the like he's basically the line. So I can set out.
Great, thank you. So. Whatever.
Yeah, I'm
All that.
You're right.
Let's start. Oh, right. Oh. Yeah.
You mean in the parking meeting?
No, like a meeting.
Yeah.
Yeah, Chuan.
Yeah, tired.
No, I said it once. No, I think. Oh, that's a different. But that's a different. From collaboration. Well, in his case,
Yep.
That is interesting.
Yeah. Oh, yeah. Fondly.
Oh,
Oh, yeah. I know partly,
my son.
from listening less than 1 hour. Pretty well. Yeah. Yeah.
I mean, they're gonna.
No, yeah, no worries. I want to find sometimes at the end.
Yes. So
For lab is. Yeah. It has, like. Substrate and non-stick perspective. Even though there is. Method that has yet been developed that provides from that just developed. Sorry. Bioengineering stuff, but like has the data and you know, you know, see that. Yeah, Maryland is, I mean, it's, you know, like everything's by their.
Maybe think about how this All right, welcome back, everybody. I hope you enjoyed the lunch and the poster session. We have two very exciting talks in the afternoon as well, and I will introduce the first speaker, Carolyn Larabell. Actually, let me just take one step back. I just realized something. There's a pattern in the talks, and so far we've been having we've been visiting the West Coast. We had a presentation from San Francisco, right? Berkeley, Los Angeles, San Diego. So Carolyn represents both San Francisco, right, and Berkeley at the same time. Two beautiful places. Carolyn is a biologist by training, but as you will see very soon in her research, she beautifully combines state-of-the-art transformative imaging and cell biology, which makes her work so exciting. Carolyn is a professor and the chair of the Department of Anatomy at the UTSF, and she directs state-of-the-art soft X-ray tomography laboratory at the Lawrence Berkeley National Lab, and runs a very, very unique resource that I think is probably unique in the world. And her research bridges both biology and imaging, addressing a very important question: how does genome look like in its natural state, or at least as natural as possible? To give you perspective, we frequently, when we look at genome structure, we slice the cell, we dice it, we perturb it in numerous ways. Carolyn is trying to ask a question: what happens if we look at the real structure without all this permutations? So we're privileged to have you. Thank you.
Is that on? Okay. Let me see if I can get rid of this annoying little word talking. I don't even see it on my screen. Oh, it looks like it's up at the top on my screen. Oh, well. On the screen, it's talking down here. Can we move that? On that one. Now it just disappeared. All of it. I'm gonna plug this back because it's by Zoom, isn't it? So yeah. My guys don't stay there. Thank you. That works. That works. Perfect. Thank you so much. Thank you. I'm just easily annoyed, sorry. So, thank you for the invitation to speak here. It's a great pleasure to be here and present to you our data. It's going to be very different from anything you've seen before. It is unique. It's what we've been developing, I guess for about the past 25 years at Lawrence Berkeley National Lab at the synchrotron. It does require synchrotron radiation. And so we're doing it here at this big round building that you see using X-rays. So how do we do this? What's it all about? I need to tell you first a little bit about how it works so you can appreciate the kind of images you're going to see. I won't get too deep into the weeds, just the basics that let you understand how we look at cells. First, what can we do with it? We're going to look at whole hydrated cells, as the dean said, in the near native state. They will be cryo-immobilized. And we're going to look at the natural contrast. There's no stains, no enhancements. It's just a differential absorption of X-rays. And we get about 35 to 50 nanometer isotropic resolution, meaning that we're looking at it all the way around. There's no missing wedge, as with electron tomography. And we're about 35 to 50 nanometer, which is not our limitation. We're limited by the optics. And so that is sort of a moving target. The light source is 2.4 nanometers. So maybe we can someday get at that level of resolution. And to image molecules, we do correlated fluorescence and X-ray tomography. So you can see the molecules with respect to the context of the cell. As I said, it's done at the synchrotron. This is the microscope itself. It's about 75 feet long. My group developed this microscope, all of it from end to end. This is the only part that the operator will see. It's this little piece in here. The rest of it is invisible. We're enclosed in a room, so you don't have to see all the tubes. A very simple microscope, really very basic. It has a light source, happens to be X-rays. It has a condenser, happens to be a zone plate optic. You can't reflect, you can't focus X-rays with glass or magnets. You put the specimen in the center, and then it has an objective that will magnify the specimen, the image, onto a CCD camera down here. This is about 15 feet, this whole thing here. These are the optics, and they're Fresnel lenses. They're a nanofabricated device made out of nickel. Just like a bullseye, the rings get narrower and narrower as you go out, and it's the width of that outermost zone that limits the resolution. At the same time, the better the resolution, the shallower the depth of focus. Whoops. So we use the 50 nanometer optic. That means all of the cell will be in focus. With the better resolution optics, which they go up to about 10 nanometers now, only about a half a micron of the cell is in focus. So you'd have to do a lot of deconvolution approaches or other ways of manipulating the image. We're working on all of those, but right now it's just easier to use an optic that gives the whole cell in focus. So we use the 50 or 60 nanometer optics. This is the really cool part of this technique, and that's how it makes an image, the absorption of X-rays. And what we're doing is we're imaging a region of the spectrum called the water window, and it's between the K-shell absorption edges of carbon and oxygen. What that means is all of the carbon and nitrogen in the specimen will absorb the X-rays roughly an order of magnitude more than the surrounding water, because we're below that oxygen edge of water. So all of the bioorganic material in the cell absorbs X-rays, water is not absorbed. So you get high contrast image, no ice in the way like with cryo-ET. But even more exciting is the absorption is linear with thickness and concentration. So the more organic material in a voxel, the higher the absorption, the higher the contrast. And that's measurable. So if you see a change in the structure with different techniques, different treatments, you can tell by the linear absorption coefficient, measurable, you can get a quantitative value for that change. So you can see it's directly linear. So when you look at a cell, here you can see the cell, this is an orthoslice, a virtual section through the cell, and it looks very much like what you'd see in TEM, But that contrast is just based on the amount of organic material per voxel. You can see the nucleus in the center and the heterochromatin around the periphery and a nucleolus there. Around here, you can see lipid drops. Lipids have lots and lots of carbon, so they're very highly absorbing. So you get a very high contrast image. Mitochondria, they have double membrane around them, and they have a lot of cristae, which is also membrane. So you get a lot of carbon, a lot of absorption, high contrast. And again, ER has less, you have lower contrast. And you can assign a linear absorption coefficient value, which I will always flip and use lac value. It's just easier. Stands for the linear absorption coefficient of that particular voxel. Now, I should have said at the very beginning, the technology is just like doing a CT scan of your knee. Think about putting your knee in the scanner. They take an image all the way around. So we do image all the way around the cell, no missing wedge. Specimen preparation, very simple. You have the specimen growing in whatever growth medium it grows in. Get a pellet of cells, put it in, put the cells in an Eppendorf, make a pellet. You can then put that pellet of cells into these glass capillaries. They're very thin-walled glass capillaries. We make them ourselves. The walls of the glass are only about 100 to 200 nanometers thick, so they don't absorb a lot of X-rays. Take it to our rapid freezer. This is a homemade rapid freezer. It's automated so that you are plunging the cell into the liquid nitrogen cooled propane at a speed faster than gravity. So it's not like your average plunge freezer. We have to make it faster than that to make the whole cell well frozen. And so we go faster than gravity, get a faster freeze. Completely controllable, the speed at which you freeze, and everything else is completely automated. You can see there's a bath of liquid nitrogen down in here, and we have these little holders called pucks with multiple little holes in them, so you can freeze multiple cells. And again, the whole freezer's automated. Press the button, it will grab your specimen, rapidly freeze it, put it in that capillary, go back, get the next specimen, rapidly freeze and put it in the holder. So you can freeze up to 90 some capillaries, then take them all down to the microscope for imaging or store them in liquid nitrogen until you're ready to image. And this is just a picture of the freezer. It's a little Star Wars like, lots of flashing lights and noises because it is computer controlled. You have to make sure that people don't hurt themselves. There's a lot of safety devices and bells and noises and flashing lights. And that whole thing takes about 10 minutes per capillary. So very fast specimen preparation, fully living state. You then take them down to the X-ray microscope and a cryo transfer device, put them in the X-ray microscope, push one button, and it will collect all the images from all the way around the cell. A projection image, multiple angles, between 90 to 360 images, depending on the size of the cell. The bigger the cell, the more images you need for a full tomographic reconstruction. It's all automated. Once all the data are collected, it's automatically sent up to our server, automatically aligned. All the images are automatically aligned with each other. You don't need any fiducial markers, and automatically reconstructed. So within 5 minutes, full reconstruction of the cell in front of you. And you can see the entire cell, all of the ortho slices through the cell, which we're flipping through here. See the nucleus popping up, heterochromatin in the center, very highly contrasted heterochromatin, and all the cytoplasmic structures around it. Completely automated. So within 5 to 10 minutes, you have your cell. That means you can get statistically significant numbers of cells. You do one after another. And also, if your treatment was good, if you have lousy looking cells, you throw the capillary away and start over. You haven't lost a week of time. This is the tedious part where you segment the structures, you manually trace them or use the lac values to get the volumes and all the other measurements you need of the structures. That's the tedious part right there. Now, what I just showed you was cells in suspension. What about adherent cells? Well, you can do adherent cells too. You take them from the flask, put them into the glass capillary. Then we put them into this little device. And these are the capillaries sticking inside of a holder. The tip is down in these 96 well plates, the plate there, and in a growth medium so that the cells are maintained healthy state. You can add whatever nutrients or whatever treatment you want to the back of the capillary. You can put them back in the incubator for a couple of hours so they adhere to the walls of the glass capillary. Then you just take them to the freezer. So same as everything you did before, you just have that intermediate step in here where you put them in the incubator and let them adhere to the walls of the capillary. Whoops, that's not what I wanted to happen. Try that again. There we go. And you then have a capillary full of cells adhering, and you can see they actually form a very three-dimensional structure. You can put collagen in there. We have collagen in these capillaries. So you have a three-dimensional structure, and they look very much like a fibroblast growing in tissue. And that is the fibroblast. So you get a very nice three-dimensional cell. Rather than adhering and forming the artificial structure of a flat cell in plastic, it's in its three-dimensional state. So that's how we do it, very simple. No single imaging technique can answer all questions. They all contribute some information. So what does X-ray uniquely contribute? How can we contribute novel information? And the one thing we have is that lack value, so that we get those measurements. We can give you quantitative information about the molecular composition of each voxel. And we can also do a lot of cells. So it's fast. You saw 5 minutes of data collection. We can image large numbers of cells. Cells are very heterogeneous. No two cells look alike. Vast range of cell structure, even from the same capillary. And so we can look at large numbers and get statistics. Those are the two things that make it really unique. It's difficult to get statistics with EM, and the linear absorption coefficient value is truly unique to this technology. It also, that linear absorption coefficient value is how we will image a lot of structures. And so it allows us to do things like segment the nucleus. You manually trace, and we're going to use a program called Amira. It's a 3D visualization program. Manually trace the nucleus in multiple sections so that you can then see the entire nucleus. You can then grab all those voxels in that nucleus and plot a histogram of the nucleus overlying the cytoplasm, the whole cell. We have the nucleus, and it has been divided into two peaks. You can see it's a bimodal distribution of the nuclear structure. And what you see that looks like heterochromatin. That wasn't supposed to happen either. Okay. Segment the nucleus. So what looks like heterochromatin and defined as heterochromatin in electron microscopy shows up in this peak here. It's a very highly absorbing end of the spectrum. Tightly packed chromatin, highly absorbing. So that's the heterochromatin peak. And you can, by just on the screen in a mirror, adjust the histogram so that you see those heterochromatin molecules, grab all of them, and then you get the 3D distribution of heterochromatin like you see here. The heterochromatin being in blue and the region euchromatin in green, and it contains the active genes plus a whole lot of other stuff. It's a very nice way of getting a quantitative distribution so you get spatial and quantitative information. You can also do things like segment the mitochondria. They have a unique linear absorption coefficient. Then you get the 3D distribution of the mitochondria. And once you have those segmented mitochondria, you have the volume, you have their spatial distribution, shapes, all sorts of information. And so you can compare volumes of structures after different treatments. And you can wrap it around the nucleus to see how those mitochondria are interacting with the nuclear envelope. Now, just because we were wondering what does the crowding tell us? So we're looking at heterochromatin. You see that it's higher absorption than the euchromatin. That means more crowded. How crowded is crowded? What is it? What does that value mean? So we looked at a yeast expressing an alcohol oxidase crystal. We forced it to express that. We know the atomic composition of the crystal based on cryo-EM. So we calculated the linear absorption coefficient based on the known concentration, how many elements are in there. And then we measured it. And it turns out that that's exactly spot on. The linear absorption coefficient is exactly what you would expect if you calculate it. So it is quantitative, but it tells you that we're less concentrated than a crystal. And sperm chromatin is the most highly compacted chromatin. And you can see the most of the cell is significantly lower compaction, less crowded than the sperm head. And if you look at it quantitatively, there's actually, if you look down here, there's a very thin blue line. That's the lipids that spread out there. So it's about as crowded as a lipid droplet, just to put it into perspective. And we've been able to identify a number of structures just based on linear absorption coefficient, as you can see here. So that helps us to segment and get some information. But what can you do with these sorts of data once you have them? We decided to look at a couple of things. The first one we did was looking at olfactory epithelial neuron differentiation, looking at the cells as they differentiate. And this is a project that we did in collaboration with Stavros Lavardis when he was at UCSF. And he was studying olfactory receptor gene selection. And what he did know is that there are about 1,400 olfactory receptor genes in clusters across 18 mouse chromosomes. So all the chromosomes have one of these genes and they cluster together. And each of the neurons expresses just one allele of one gene. So it's a highly selective process during differentiation. And that process all occurs during neurogenesis. And so that's what we wanted to look at, the organization of the nucleus during that process. So we got epithelium from mouse. These are all cells that we obtained from mice. And Stavros had markers for the different layers. So you can see that stem cells are at the bottom and they differentiate as they go up. And these are their epithelial cells at the top. So he has markers for each of them. We were able to get cells that we knew were at different stages of differentiation and study them. And so what we see here is those cells, the nucleus on the left, you can see the organization here highlighted in red, which I think you can see, and the histogram of that nucleus. You can see there's a lot of heterochromatin around the periphery. You can see the histogram and the 3D distribution of heterochromatin and euchromatin looks fairly normal. And this is showing you the distribution of that chromatin in the outer 2 microns of the nucleus. Outer 2 microns of the nucleus, the euchromatin and the heterochromatin. So a lot of heterochromatin at the periphery. What you can see happens during differentiation, though, is that the heterochromatin starts leaving the nuclear envelope and moving towards the center of the nucleus. And you see nice mass of pericentromeric heterochromatin, which you heard about earlier, a mass of pericentromeric heterochromatin, and a nucleolus at each end of that mass of chromatin. You can see the change in the histogram as the cells differentiate, the change in the spatial distribution. The 3D shows very nicely that mass in the center and very little around the periphery. And you can see it when you plot it. So you get both quantitative and spatial distribution of the chromatin during differentiation. Now, one thing Stavros noted when we did this is this little blob down here at the periphery of the pericentromeric heterochromatin. And that made him very curious for a reason that he had observed for many years and didn't really understand put in place. But that made him wonder if those were the differentiated, the silenced genes. And the reason he wondered that is rather historical during his grad school days. But to prove it, he made a marker, a pan OR marker that would recognize multiple olfactory receptor genes. And with fish, it showed that indeed there are foci around the pericentromeric heterochromatin of those silenced genes, about 5 or 6 of these foci. And it does indeed match this focus here. And then one allele will pop out of the silenced genes and be the expressed allele. Stavros has done many beautiful studies to really go into the details of all those processes. But for our purposes here, we're just going to focus on this one process. What he had noticed previously that piqued his curiosity was that as the cells are differentiating, and you can see them here, this is a DAPI stain, you can see that there's the increasing accumulation of that pericentromeric heterochromatin in the center. So he'd always noticed that and been curious about it. And then he did a lamin B stain and showed that epithelial cells have a periphery of lamin B, as you would expect in the nucleus, but the mature cells do not. In their last stage of cell cycle, the last time they divide, the lamin B stays in the cytoplasm, leaves the nucleus, And that allows that heterochromatin to move to the center. So it's a complete structural reorganization. No lamin B, paracentromeric heterochromatin goes to the center of the nucleus. And those silence genes accumulate around the periphery. So what he thought was, is it necessary for that lamin B to leave? Is it necessary to have that mass of paracentromeric heterochromatin? So he got a mouse where he forced it to express lamin B inappropriately in the final cell cycle, in the final division. And what happened is that the mass of paracentromeric heterochromatin is gone. It's sort of a little bit up here, forced to the side. You see a nice collection around the periphery of the heterochromatin. And indeed, all OR expression is screwed up. So in this particular cell line, the 3D organization of the nucleus is critically important for gene expression. You have an improper organization, you screw up all gene expression. He actually has gone on to further these studies and showed that they want to isolate these cells and grow them in culture. And so he started growing them in culture. It took a lot of work. They had never been grown in culture before. He finally got it to grow in culture. But what happened then? It's the same thing. The entire thing flattened out, loss of the central mass of paracentromeric heterochromatin, and completely screwed up gene expression. So the 3D organization of this nucleus is critical for proper gene expression. He also knocked out HP1 beta to see what would happen, and that also is critically important for the proper gene expression. And you can see in here the distribution of chromatin, euchromatin, and heterochromatin is completely disrupted when you knock out HP1 beta. So 3D organization is critical. We then looked at chromatin condensation during hematopoiesis, blood cell differentiation. And we started looking at early development. As you know, maybe you don't, blood cell has two lines from the pluripotent or multipotent stem cell. It can become either the red blood cell, white blood cells, or progenitor cells, actually not red, just the lymphoid or myelocyte lines. We looked at the stem cell, we looked at progenitors and two differentiated cells. You look at, we started actually with an embryonic stem cell to get sort of a baseline of what the pluripotent cell would look like. And you can see this cell has very large nucleoli. Those red structures that you saw highlighted are the nucleoli. It's a very large nucleus, very little heterochromatin around the periphery, mostly euchromatin in the center. So that's the embryonic stem cell, very large. Blood cell goes down to here, and you will see a much smaller nucleus. Then we looked at the progenitors. We'll look at one progenitor, much smaller cell, round, much more heterochromatin in the periphery, smaller nucleoli. It's already showing signs of distinct differentiation. And then when you look at the differentiated cells, the B cell or a neutrophil, we're looking at the neutrophil here. You see a very thick band of heterochromatin around the periphery, small nucleoli, and a lot of euchromatin. Neutrophils are very weird, though. They have a very interesting looking nucleus. If you look at the 3D organization of the nucleus, it's sort of toroidal, very unusual, unique to the neutrophil, but makes a very interesting looking structure. If you do the quantification of this, you can get more specifics on how the heterochromatin is changing. You can see this is the stem cell, very little heterochromatin around the periphery, much larger nucleus, predominantly euchromatin region. You're getting more and more heterochromatin around the periphery in the progenitor cells, and then in the differentiated cells, nice thick band of heterochromatin around the periphery. Again, this cells from mice, so they're not cultured cells, they're right from the mouse. And you can quantify it and see that there's a distinct increase in the heterochromatin during that differentiation, decrease in euchromatin region, and a very distinct decrease in the cell volume, nuclear volume, get smaller. It's a little bit easier to see the bands of heterochromatin here, and you can plot it, and you see that the band of the differentiated cells have a much thicker band of heterochromatin than does the stem cell or undifferentiated cell. And so you get both spatial and quantified information. Now, one thing we noticed in both of these cells was we're seeing that the cells get smaller as they differentiate, but we also noticed that the euchromatin region is more directly proportional to the volume of the nucleus. In other words, as the nucleus is getting bigger, it's mostly the euchromatin region rather than heterochromatin. So you're getting more proteins, maybe more active genes, a lot more stuff in the nucleus. So we wanted to now look at primary cells versus transformed cells. We had a lot of information about B cells from mice. If we thought we'd look at cells from mice for the next study, let's just flip through the orthoslices of the primary B cell. You can see a large amount of heterochromatin at the periphery of the B cell nucleus. And now we're going to look at a B cell in which we've transformed it, only transformed it using v-able. And you're going to see a very different structure of the nucleus just based on that transformation. Nucleus is much larger, thinner band of heterochromatin around the periphery, much larger nucleoli. It's already a very different cell just from being transformed. Then we look at the lymphocytic leukemia, the same cell line, the same B cell from mice, lymphocytic leukemia, massive nucleus, almost no heterochromatin around the periphery of the nucleus, gigantic nucleoli, a very different cell. You can quantify it, and you can see the increase in the nuclear volume from 102 cubic microns up to 450 cubic microns, and the percent heterochromatin from 52% down to 22%. It's a huge nucleus, but mostly the euchromatin region. And what that means is that band of heterochromatin becomes very thin around the periphery. Now, 3D organization is important. Heterochromatin at the periphery is very important. You've completely altered the organization of that heterochromatin throughout the entire cell. And again, though, with these cells, the euchromatin region is directly proportional to the volume of the euchromatin of the cell. So periphery, the bigger the nucleus, the more euchromatin region you get. And we've gone on to do other cell types. You will see that the more you get very large cells, nuclei, the MDCK2 is getting up to the 400 cubic micron range, very large. And with differentiation, you get a distribution, a different thickness of the heterochromatin at the periphery. The lymphocytic leukemia cell way down here compared to the primary B cells. So my theory is, and these are very crude drawings, that this is sort of what they look like. Differentiated nucleus, very round, very small, thick band of heterochromatin. Both stem cells and the dedifferentiated cells, cancer cells, all look alike. Very thin band of heterochromatin, very large nucleus, they look alike. The nuclear organization, you can't tell them apart. So go back to this slide for just a minute because of the size of these things. We have looked at thousands of cultured cells in the last couple of years, literally thousands. All of the cultured cells look the same. The more cancerous they are, the more they look just like this. Very hard to tell one from another. The moral of the story, I think we need to get to more cell models, perhaps organoids. We have to start looking at more normal nuclei than what you find in a cancer cell, or even this is what cell lines look like. No matter what the cell line, how normal it started out, they all end up looking like this. So they become pluripotent. They're not pluripotent, sorry, a lot of polyploidy, aneuploidy, multiple chromosomes, they are no longer normal. We need to start looking at more cell models. So leave that aside. What we wanted to do was look at a cell line, human cell line. Everything we've done so far was in mice. We wanted to look at human cells, but we wanted them to be as normal as possible. So this GM line is available, and they guarantee it to be normal when they send you normal number of chromosomes, perfectly diploid when you get them. What you do with them over time, the more they grow in culture, the less normal they will be. So we looked at very early passage. You can see this looks much more normal. Nice band of heterochromatin at the periphery, nice distribution of heterochromatin throughout the nuclear or throughout the entire nucleus. A very nice looking healthy cell. Just look at different orthosplices throughout. Nice nucleoli, nice nuclear organization. It's a normal cell. It's much more normal. So I promote the use of cell models and organoids and culture and biopsies, et cetera. Anything a little more normal than what we get in cultured cells. They're just what allows them to be so easily transformed, which is why people like to use them. They're no longer normal. So that's my lesson there. But what these lac values allow us to do is things like model how molecules move through the nucleus. So this is a study done by a mathematician. We work with multiple people, and he wanted to model how a molecule would move through the chromatin based on the concentration of stuff in it. So he made a protein of a known size and a potential binding site and started his model. And you can see in this piece of a wedge of a nucleus, the multiple paths that it would take. And what he found was that there's an optimal crowding for molecules to move through the nucleus. So it needed to have that optimal crowding that allowed it to move through the nucleus and find its target. Another modeling based on those same B cells, these are all the same cells. This was done with Frank Albert, and what he did was take the same B cells, he did the high C studies, then he modeled how that genetic material would have to fit into the nucleus to match the high C studies. And he found one of the patterns that he saw was 3 clusters of centromeres based on the paracentromeric heterochromatin. And he asked us to do the SXT, the X-ray tomography of those same cells, which we did, and we were able to find that same pattern. So in this case, the high C, he modeled so it had to fit into that nucleus. He was able to match one of the structures that we see, the 3 different centromeres and the heterochromatin around it. So what we'd like to do eventually is start doing the reverse, image a cell and then do the high C of that same cell. So it requires some technology to work out, but that's something we can do. So that's some modeling. The bacterial nucleoid is again where we looked at the differences between the supercoiled domain seen with a lac of 0.29 and the lower lac, and how it responded to acid stress, and the SAC study they did to show how the molecule would change organization to generate the nucleoid structure that you see in the lower right in the acid stress nucleoid. Kind of a nice model, very simple structure, and that is the nucleoid. Chromatin connectivity. What we did here was look at the distribution of heterochromatin. The old theory of islands of heterochromatin is not accurate. If you look at this movie, you can track the blue and the green, and it's continuous throughout all of the nucleus. You can do a shrinkage study of the chromatin, and the heterochromatin will shrink down to a structure where 99% voxel connectivity. You can shrink down the euchromatin. You're basically just shrinking the size of that structure as you see it in the voxels to a minimum structure and find that it's all interconnected. That's that massive paracentromeric heterochromatin. These are Stavros' cells. And then the overlay. And so you can see that there's a chromatin network, some sort of baseline that we see in the nucleus. And we see it in every nucleus. We cannot break that basic connectivity. Don't know what it is, why it's there, what purpose it has, but it's there. Membranous organelles. I'm going to visit Gary's favorite topic, the nucleolus. And this is the nucleolus from the mature olfactory epithelials we saw. And it has the 3 structures that Gary described. You have the central structure, a band around that structure, the 3rd band out there, and then the paracentromeric heterochromatin. They are all, as he said, separated based on no membrane, but very nicely separated and distinctly seen. And then this is the 3D distribution. This is a video, the paracentromeric heterochromatin around the periphery, and the 3 structures with them. Now, what we thought, based on reasoning of crowdedness, based on the lac values, that region right there, where it looks the whitest, is the lowest density So is that where transcription occurs? That would be logic telling you that's where transcription would occur. So Stavros did the fish study to show, and this has to be a separate cell because fish is very destructive. You can't do structural studies on a fish treated cell. But taking the same stage of differentiation, you could see, oops, you could see the rRNA, sorry, in green, in that low transcription region, the nucleolin in that central one. So we've hypothesized that is where transcription would occur. We did a transcription factor, and indeed it does overlay with that region, low absorption. So far that seems to hold up. That's where transcription would occur. And we used that to hypothesize where transcription sites would be out in the rest of the nucleus. It all showed up in the Euchromatin region. You see those little red dots. That's the nucleus bump there, nucleolus, I mean. And so we got all those low absorbing regions in that site right there. This is a different stage of that very same olfactory epithelial study that we did. This is this is a differentiating cell, so it's a little more active than the mature one that you looked at before. And in addition to those three regions that we saw, we also see these very dense deposits moving through that outer region that Gary described where the transcripts move. Based on just pure logical reasoning of the density of those structures, that they might be the RNA transcripts. We need some direct evidence direct overlay to show that, but it fits with reason. Number of these deposits only in that region out there and in the differentiating cells, not the mature cell. Virus infected cells, I don't have too much time to go into it, but we're able to differentiate the capsids versus the enveloped virus on the surface, capsids in the nucleus, and some on the surface. And then I want to show you the model. All the structures we can see in the infected cells, virus infected cells, but I want to show you the video. This one shows how a virus particle, the capsid, would move through the nucleus, another modeling experiment. And so they modeled how 1,000 capsids would move for 5 seconds and showed that their size restricts their diffusion into heterochromatin, limits their movement through the euchromatin region through the nuclear pores. So just with these images we have, we can do modeling based on knowing the concentration of the material things are moving through. I have to flip through the SARS because I think I'm out of time. We did some SARS infected cells, what we did during COVID vacation, and the only experiment they allowed at the synchrotron. So we looked at SARS infected cells. But the one thing we found that's really interesting is that a lot of the infection occurred by two cells fusing with each other, and they transfer the infection via that fusion, and you end up with a binucleoid cell. We had a lot of those double nucleus cells in these SARS infected cells. Quickly go through some quantitative imaging because we can look lots of cells. We looked at insulin granule secretion. You can do a bunch of manipulations and count the number of insulin granules. Lots of statistics from those cells. But another cool modeling experiment, because I think this tool is really good for cell modeling. This was based on some work done by Art Olson and David Goodson. They took the proteome from 20 known proteomes of the insulin granule itself, and they had this crystal structure of it. And so what they then did was model how those proteins, based on their crystal structure and our lac values, would fit into the insulin granules in an immature versus mature insulin granule. And you can see that the low lac values for insulin granules correlated with their immature, and the high lac correlated with the mature granule. So, I mean, this is a really, in my opinion, a really cool study combining proteomics and structural imaging, because it wasn't trivial, but they used that proteome and the lac values and came up with a model that worked. I like it. And then just David Goodson does all the models for cell biology textbooks. This is his model of insulin granules, immature and mature granules. Correlated imaging, just quickly, we do correlated fluorescence and X-ray tomography. This one shows the inactive X chromosome based on that H28 EGFP that preferentially binds the inactive silenced chromatin. And you can see that we can overlay those data from the cryofluorescence tomography and cryo X-ray tomography and get a 3D view of that chromosome based on the linear absorption coefficient. You can see that there are 3 different degrees of compaction in that chromosome based on linear absorption coefficient. Lots of studies that could be done with that. We can also do correlated correlated X-ray tomography in cryo-ET. Not the same cell, but this is an example of insulin granule again, where we were able to count numbers of insulin granules, changes with various treatments. And with cryo-ET, you can see the structure of that insulin granule. 2 very compatible tools. We get to see the whole cell, do the quantitation. They get about less than 0.1% of a cell, but you can get the high resolution information and get more information from 2 technologies than you would get from either one alone. I'm not going to go through automated segmentation. It's something we need to do. Painful. Got too much stuff here. Protein condensates. Ending up with the Lewy body condensates. So we're also able to see all of these condensates, membranous organelles, whatever, and starting to look at Alzheimer's condensates and Lewy body condensates. Stop there and thank all the people. Our lab here, Mark Legros is a physicist who designed and built all of our instruments. These are the people who do the work, and our wonderful collaborators who bring exciting and interesting specimens to us for imaging. And I will stop there because we've run out of minutes left for me. So
Thank you so much. Well, I think we just had a very clear example that looking in 3D with as little disruption as possible really gives you very unique insights.
yeah, very different types of
Very interesting. Very interesting. All right. We have one more talk today, and I would like to introduce Yogesh Goyal. He's a rising star here at Feinberg at the Department of Biochemistry and Molecular Genetics. He was one of the pioneers who developed the concept of rare cells and brought very powerful, innovative understanding of how cancer cells may develop resistance. He will introduce the next speaker.
Thanks, Vadim. My name is Yogesh, and I just started here at Northwestern last year, and it is my pleasure to introduce Chuan He. And I'm sorry, but I'm breaking the pattern, and we are moving towards Midwest and Chicago. So Chuan is a John T. Wilson Distinguished Service Professor at the University of Chicago in the departments of chemistry and biochemistry and molecular biology. So Chuan did his undergraduate in the University of Science and Technology before moving to MIT for his PhD, and he stayed there across the river to do his postdoc at Harvard. Soon after that, Chuan started his lab at UChicago and very quickly rose through the ranks to become a full professor in 2010. And soon after that, he was awarded the HHMI investigatorship. And his work has really transformed how we think about transcription and gene regulation happens through reversible methylations. And his group, in fact, made a huge discovery which showed that it's RNA reversible methylations that can control and regulate gene regulation and mRNA translation. And the group has since gone way deeper into a lot of these processes, really finding finer details of initiation and maintenance. And one striking feature of Chuan's work is there's really no boundaries. I feel like the lab is really combining biochemistry, cell biology, RNA biology, epigenetics, and now genomics to get at these questions and really new exciting technologies and concepts all throughout. So I'm very excited to welcome him. Take it away.
Bye.
All right. I'd like to thank Vadim for this opportunity. Make sure I can move this. Yeah, my lab works a little bit on the chromatin structure, but I don't feel like that's our expertise. So instead, today I'd like to actually share with you something related to chromatin regulation. So our work actually on the RNA modification side. And many years ago, we proposed this idea that some of these RNA modifications might regulate gene expression in ways analogous to DNA histone modifications. And in fact, we were that was many years ago. We were working on DNA demethylation, and we wondered some of these DNA damage repair enzymes could actually reverse RNA methylation, and that actually led to a discovery of a RNA demethylase. The reason I want to use this as an introduction slide, as you can see in a few minutes, we'll come back to this because back then we discovered this as a messenger RNA M6A demethylase, and turns out it also works on the chromatin and regulates chromatin structure. So back then, the substrate we discovered for this reversible methylation are N6-methyladenosine, and it was actually well known to occur on mammalian messenger RNA. It was discovered back in 1970s. It's essential for most human systems, and if you calculate, it's about 3 per messenger RNA. So there's a writer component. People know for several decades, we came in and showed the process is reversible, at least on the messenger RNA side. And also we showed the methyltransferase when others are actually very big, 1,000 KD. So to my knowledge, it might be the biggest in any DNA or histone modification systems. And on the messenger RNA side, I'm just going to give you a few slides before I transition to chromatin state. We and others spent almost 10 years map out the detailed pathways, how this modification would affect post-transcriptional regulation. There are these reader proteins that recognize hundreds of methylated messenger RNA, and they were affected their decay. Some of those were actually stabilized, and others were mediated translation regulation. The sort of future of these the RNA methylations is the modifications essential to most cell differentiation development. If you knock out the methyltransferase in embryonic stem cells, the embryonic stem cells stuck at a very early stage, right? Mouse died at the early embryo stage, but the stem cells were stopped differentiation. That's probably the case. That's actually turned out to be the case in many cellular systems, pro T cells, naive T cells, pro B cells, neurogenesis. It doesn't matter what systems you work on, the methylation is essential or critical to cell differentiation. Now, one sort of just to give you one example, the reader proteins of YTGDF2 mediate decay of these messenger RNAs, and we previously suggested that what they really does is mediate this transcriptome switch. So whenever you have a cell, you want to differentiate to the next stage, you have the clear transcripts that dictates the previous cell state, and that's actually goes through the methylation and sort of coordinated decay of these transcripts. And examples are, if you look at maternal to zygotic transition, neurogenesis, or even look at stem cell expansion. If you get rid of YTGDF2, you impair the cell state transition, and you can dramatically expand the hematopoietic stem cell in this case, but also other stem cell systems. So we had a lot of fun, and the field's been moving forward. This has been recognized as one of the key mechanisms or pathways to regulate mRNA stability. In 2008, we started to realize if you knock out the methyltransferase, the phenotype is turned to be a lot more severe than you knock out the individual of these reader proteins that recognize the methylated RNA. We also started to realize that some of these methylations, if you sequence the whole sort of RNA, and you realize that some of these methylation seems to occur on non-coding RNAs, and they actually align well with these promoter-associated RNAs, enhancer RNAs, or even the ritual transposon RNAs. So we wondered, in addition to a post-transcriptional regulation, whether some of this methylation actually regulates transcription, or further, maybe they can somehow factor the chromatin state. The first experiment we did, we actually directly asked Howard Chuan for this mouse embryonic stem cell, which he made with depletion of MET203. This is the key writer component. And later on, we made our own. And we did a few very simple experiments. Immediately, we realized that if you look at the EU labeling, the total nascent RNA output increased quite dramatically. If you perform a tunnel analysis, you can clearly see the chromatin become more open in this when you deplete MET203. You can complement with a Y-type methyltransferase, but not an inactive mutant. So what's going on? We quickly move to sequencing. We can isolate the chromatin fraction, obtain all the RNAs, deplete the ribosome RNA, and the sequence M6A methylated RNA. What we realized is, of course, we saw depletion of methylation on messenger RNA or pre-messenger RNA, but we also see depletion of these methylations on enhancer, promoter, repeats RNA. Interestingly, without methylation, these RNAs show higher level on the chromatin, all right? So their levels on chromatin increases. Somehow the methylation suppress or reduce the level of this RNA on the chromatin. We turned on to another nuclear reader. This one is also essential, actually very, very important. You knock out mouse embryonic stem cells, even that shows some defect. Mouse died at a very early stage if you get rid of YTGDC1. We observed a very similar thing. We can see more nascent RNA production when you get rid of YTGDC1. You can rescue with a Y type, but not a mutant that would not bind methylated RNA. And this is consistent with tunnel analysis. So at least the simple hypothesis, some of this chromatin RNA can be methylated, and a portion of those might be recognized by this reader protein, which actually we know previously is adapter for nuclear exosome complex that would decay RNA, right? So that leads to decay, and perhaps that affects nuclear or the chromatin state. So you can knock out YTGDC1 and perform sequencing of chromatin RNA. And in this time, you see a little bit of change on enhancer promoter RNA, but majorly you see this repeats RNA. You really accumulate this repeats RNA when you get rid of YTGDC1. The major types of these repeats RNAs are line RNAs, ERV, and other ritual transposon RNAs. In fact, if you follow the normal decay, this is the normal decay of the line RNA, you can see this, but if you knock out methyltransferase or you knock out YTGDC1, you delay the decay of this on the chromatin, okay? So essentially you have this mechanism, you can methylate the RNA, and particularly the ritual transposon RNAs can be decayed through YTGDC1. What's the biological consequences? How is this related to the chromatin state change we just observed? It turns out, if you perform sequencing, there's something like 6,000 genes that have upstream methylation or methylations occur to the introns, the ritual transposons. And if you get rid of M6A, 99% chance you see increase the transcription rate of this downstream transcripts, right? Nice, 6,000 transcripts, that's a major effect. If you look at the chromatin state or look at histone marks there, if you get rid of methyltransferase, you accumulate K4 trimethyl, K27 acetylation. And if you just perform ChIP-seq experiments, look at the transcription starting site. If you get rid of methyltransferase at this 6,000 genes, you always see increased K4 trimethyl, K27 acetylation, indicating a more open local chromatin state, right? So not just globally you see this, but then these methylations indeed appear to regulate downstream gene transcription and the chromatin state. What's going on? Well, I don't need to show these papers, where we always thought these RNAs are spaghettis. They probably are, but they're quite important in whatever condensate formations discussed today, or to me, a symbol of transcription active complexes, right? And they're there to help assemble transcription active complexes. And we believe the way to regulate this process is probably through modification, right? There are several ways to regulate this. For ritual transposon RNAs that help regulate the nearby transcription, you can methylate and decay that piece of RNA. And later on, other labs show that the presence of the methylation is critical to recruit histone modifiers to install stuff like K9 trimethyl, right? And that way leads to repressed chromatin states. Now, everything is correlation at this moment, right? What about the causal relationship, right? To answer that question, we quickly put together a small CRISPR system. This is the RNA targeting dCAS13 inactive CRISPR, which fused to a demethylase, so we can perform site-specific demethylation. So we picked the 10 loci, and here's the one. There's just one example. We delivered this specifically to each methylation site. Here's the super enhancer RNA. There's some methylation. If you knock out the methyltransferase, you see the methylation is pretty much gone. And you look at the K27 acetylation, you see a dramatic increase, right? So the methylation correlated with low K27 acetylation. Depletion leads to higher K27 acetylation and more or higher transcription rate downstream. If we deliver this towards that particular methylation site, we can reduce methylation on that enhancer RNA. We increase the half-life time of that enhancer RNA, and we increase transcription rate, K4 trimethyl, K27 acetylation. So we did this to 10 different loci. At least 9 loci showed consistent results. So with that, we pretty much established this new layer of regulation. We believe there's this methylation occurring on non-coding RNAs, and that appears to play quite important roles. You're going to say this is just a part of the story. A year later, 3 labs independently made very similar discoveries, and 2 labs showed some of the really nice pictures or videos, images this morning. There are actually 2 labs showed that the methylation is essential to reestablish heterochromatin during mitosis. There's some really nice slides that they showed, right? Some of the heterochromatin is gone and the transcription occurs. We don't know that's a aberrant transcription or transcription related to regulation. Once you have transcripts there, they needs to be methylated to recruit proteins, reestablish K9 trimethyl. If you don't have methylation, you can't reestablish heterochromatin in those regions after mitosis, right? So I encourage you to read these papers. All right, so unfortunately, when we talk about chromatin state, we can't just talk about DNA modification, histone modification. So we somehow, unfortunately, add this another layer of complexity on these non-coding RNAs. And I showed you the writers, the readers, right? We have methyltransferase that seems to mediate methylation, right? And then the readers that comes in seems to affect the decay or recruit histone modifiers. But DC1 only works on retrotransposon RNA, right? I showed you there's enhancers, promoters, right? It can't be just the one reader protein, right? Mediate this regulation. So naturally, we went ahead and look at the ENCODE data, or we look at the RNA binding protein CLIP-seq data correlated with chromatin RNA M6A sequencing. And we look for overlap of RNA binding versus M6A sites. And a few proteins show up. RBM15 is actually a component of methyltransferase. K36 trimethyl is well known to recruit methyltransferase to facilitate the messenger RNA methylation, and the POR2, obviously POR2 is important. And then we have RBM22 and RBFOX2. So we give our friends RBM22 to work on. We decided to focus on RBFOX2. Because this is actually interesting splicing regulators, has an interesting conserved motif. The bizarre part of this is that it has a phenotype, quite dramatic phenotype, but this phenotype is not related to the splicing defect. People know that, right? It's a well-known splicing factor. In my mind, lots of nuclear RNA binding proteins being labeled as splicing factors, probably right so, but many of those probably have additional functions. This is one. And it shows good overlap with M6A. And very interestingly, this thing has 2 distribution profiles. Within genes, indeed, it's close to the exon intron splicing regimes. It's there to regulate the splicing, but a portion of this locates to the promoter sites. All right, it's not within the. It doesn't attach implants. It's really at the promoter sites. Now, if you zoom in into the promoter sites, they overlap with M6A extremely well, right? And then when we use a methylated probe, perform a pull down, we enrich this protein, and we pull down the protein with this binding RNA, and we enrich M6A, and we can sequence that RNA. We obtain the exact methylation motif, right, under the protein binding sequence, and we use that motif. We methylate it without methylation. We perform pull down again. We can enrich RBFOX2. So all these results indicates it binds promoter RNA and selectively binds methylated ones, or preferentially binds, I should say. It's a lot of sequencing experiments you can ignore. I just want to show you that the depletion caused the hypomethylation, open chromatin, gene activation, and all this occurs with RBFOX2 and M6A. If you have RBFOX2 and M6A there, you see the major effect, right? The increase of K4 trimethyl, it's a suppressor, right? If you get rid of it, at the M6A side, you see the most open chromatin state. And also it interact with one of the component of methyltransferase RBM15, and only when you have both RBFOX2 and RBM15 binding, you see again the most dramatic effect on M6A. Mechanistically, we did a lot of experiments, and previously people thought RBFOX2 directly binds PRC2, and what we showed is actually RBFOX2 most likely binds YTGDC1, and then remember it's the same YTGDC1, and this YTGDC1 recruits PRC2 to add a suppressive sort of marks, right? So this is the way we believe, almost like a translational factor, but it doesn't bind DNA. Instead, it binds that methylated RNA to mediate nearby K27 trimethylation to facilitate the suppression of gene expression. What's the biological consequences? RBFOX2, if you look at the hematopoietic stem cell differentiation, the level comes down. It seems to suppress hematopoietic stem cell differentiation. And that's indeed the case in collaboration with Professor Jianjun Chen, the City of Hope. We knock down RBFOX2 in variety of different systems. You can see that leads to, that seems to drive differentiation in this myeloid system. And in leukemia, if you drive differentiation, obviously the first thing you think about is the leukemia systems. This is a well-established systems. When we knock down RBFOX2, we seems to promote leukemia differentiation. We seems to prolong survival of the mouse. I don't think this has any therapeutic impact at this moment, but it's actually interesting to see this is one system, and we're actually just obtained the second system in a very different, it's not even a blood system. We found RBFOX2 really regulates stem cell differentiation. In that system, there might be therapeutic potential. Mechanistically, I personally believe this is all these are group effect, but whenever you write a paper, reviewer wants to say one gene. So we say, okay, here's a TGF data everybody love, right? This one has a promoter M6A right there, and is actually bound by RBM15. When we perturb RBFOX2, we can see M6A change, and we can see PRC2 binding difference. And I believe we even delivered a demethylase specifically to that side, and we can show we change TGF beta binding. So at least in one cases, this system seems to suppress TGF beta through M6A, through this chromatin M6A mediated regulation. All right, so I think what I showed you is one reader that touches upon the ritual transposon. And now we have another reader, which seems to bind promoter RNA. And I fully anticipated there are additional proteins that may work as transcriptional factor, but instead they bind methylated RNA. I want to finish with my last story about the erasers. I told you the writers, the readers, or the M6A binding proteins, but also the process that can reverse this on the chromatin. This goes back to the introduction. The first protein that got us into this field is this FTO, which mediates demethylation of M6A. We did this in many cancer cells in early days, oxidize the methyl and remove the methyl group. And this is the HeLa cells. If you isolate the messenger RNA, if you get rid of FTO, you see about 15–20% increase of methylation on messenger RNA. In leukemia, 40% of total methylation on messenger RNA could be reversed by FTO. Right? The phenotype of FTO is kind of bizarre, leads to late embryo death. Right? And if you look at the human or mouse tissues, except adipocytes and neurons, in many other tissues does not seem to touch messenger RNA. There are studies by us, by others, the correlation studies. It's there, but it doesn't seem to work on messenger RNA, except in adipocytes and in neurons. In neurons, it really work on messenger RNA. Right? So what's going on? Right? This puzzled us for a decade. We didn't have an answer until after we realized the methyltransferase not only install messenger RNA methylation, but it also installs this chromatin RNA methylation. We call them chromatin-associated regulatory RNA now to be different from coding transcripts. And we wonder if the FTO doesn't work on messenger RNA, maybe it work on these chromatin RNA. So very quickly, we constructed the FTO knockout the mouse embryonic stem cell. And when we isolated the messenger RNA and look at their methylation, we didn't see much. There's a tiny bit increase when we knock out the FTO, so they probably work on some messenger RNA. But if we took chromatin RNA, deplete the ribosome RNA, you see a very nice increase, about 10–15% increase of total methylation. The postdoc was very encouraged, went ahead to perform the sequencing, and indeed, what he found was line and ERV, the retrotransposon RNAs are the major substrates. When you get rid of FTO, you see M6A accumulation on these retrotransposon RNAs, in particular line 1. I'm going to just use line 1 because it's the major one. The presence of methylation leads to reduce the level of these guys on the chromatin. You know this very well. With 400,000 or 500,000 copies of line 1, they're young ones, right? The recent ones, active young ones, those are the ones mostly affected by FTO. To confirm this, we did a bunch of experiments. We isolated the line 1 and look at the M6A and the level. Clearly, if we get rid of FTO, we see increased M6A on line 1 and reduce the line 1 level. When we put on FTO, we enrich line 1. And we also applied the small molecule inhibitor of FTO to show this is activity dependent, right? And lastly, we can block line 1 with ASO and compare with FTO knockout. The expression profiles quite similar, right? All indicating the main pathway of FTOs probably goes through line 1 regulation. On the chromatin state, if we deplete FTO, we accumulate M6A, you can clearly see that we drive a closed chromatin, globally closed chromatin. If we deliver a CRISPR system with FTO targeting line 1, we can partially rescue that. So we did a fun experiment. We add FTO inhibitor into mouse embryonic stem cell Y type, and we follow the kinetics. You can see the buildup of M6A on line 1 that leads to reduce the line 1 level, and you can see reduce the chromatin state, right? Quite a nice correlation. So all this suggests what we have is probably a cis regulation. These line RNAs are M6A methylated, and FTO somehow constantly remove some of this M6A to maintain transcription active state. But if you get rid of FTO, these M6As will recruit YTHDC1, leads to repress the chromatin state. Mechanistically, I'm not going to say too much. That turns out to be 10 supporting figures in the paper, just like everybody else. But I don't know, I can't say I fully understand this. This is just observation. We don't quite understand what's going on. What we observed is that There's lots of line ones. There are line ones outside of the genes, line ones within the genes. Line ones within the genes give us the major effect. About 1,400 genes actually don't know this in mouse embryonic stem cell with line one either in introns or antisense. These are the ones affected the most by FTO, right? If you have M6A, it either leads to decay or recruitment of SETDB1. Here's the results. If you compare this line one containing genes versus the rest, if you knock out FTO, you get the most downregulation, right? So there's some local effect on really change the chromatin state through RNA methylation. Functionally, probably everybody knows the importance of line one, I assume, right? If you block line one with ASO, your 2 cells will not be able to propagate to the 4 cell, right? That important. And if you take a mouse ES cell, you can differentiate it. The line one level goes up 6-fold, FTO level goes up 3-fold. You get rid of FTO, line one level can only go up 3-fold. And that leads to increased differentiation, reduce the proliferation. And we can actually rescue this about 60% if we deliver FTO specific to line one. So line one play important role, but there are probably other ritual transposon RNAs play similar roles, not as important perhaps as line one. If you look at the mouse early embryo, we did this through collaboration with my former coworker and her previous PhD advisor. So if you look at the early embryo, if you knock out FTO, you see exact same set of genes show change, and these are same genes we discover in mouse ES cell. They have line ones within genes, and they similarly displayed reduced transcription. And if we deliver this CRISPR system into early mouse embryos, we can rescue gene expression. So I spent all this time showing you, hope I convinced you, we do have a reversal process on the chromatin RNA, and that seems to have an important impact on the chromatin regulation. Now, if you look at the mammalian tissue, this is actually a very gratifying correlation we previously could not see. If I look at the FTO level versus total messenger RNA M6A level in all tissues, I did not see a correlation. That bothered me for many years. But now if I just take a line one, I only look at the line one RNA total cell, I see a very nice correlation. The tissues with high FTO tend to have low M6A on line one and high line one RNA level. Very nice. So this again confirmed that line one is the substrate. If I look at the second demethylase discovered by my lab, this is the messenger RNA, demethylase RPH5. You see a nice correlation with total messenger RNA methylation, but you don't see this on line one. Lastly, FTO is highly expressed in heart and the brain, so we made a cerebellum specific knockout. We can clearly see increased M6A on line one, reduce the line one levels, etc. Collaborators working on the physiological importance of this regulation. So what I showed you during pandemics, all right, we were able to actually map out this additional layer of regulation on the chromatin. This is completely, well, not completely, this is mediated through RNA, but actually lots of this are going through the histone modifications, right? RNA as a way to either decay that RNA or recruit histone modifiers. And we have writers, readers, but also erasers in this whole spectrum. Because I still have probably 5 minutes, I share with you some fun stuff, all right? So we've been starting to manipulate this. We had a lot of fun manipulating in mouse. We can actually grow mouse bigger or smaller, depending on what you want. We have more mice, pups or less, etc. And my former postdoc had a much better idea. And she decided to put a human or mouse FTO into plants. I thought this will never work, but actually worked. And if we put into rice, we of course tried many plants, and rice gave the most dramatic phenotype. You see the expanded root compared to the wild type. And in greenhouse, we saw 3-fold increase of biomass and yield, right? The best people have ever done in the past is about 20–30% with heterosis, hybridization, right? We can do 3-fold in greenhouse. In real fields, under 2 different climate conditions, we achieved the 50% increase. These rice with longer roots are more drought resistant and resistant to other stresses. We were bothered by mechanisms for years until we worked on mouse embryonic stem cell, and we learned what we learned there. We come back thinking, we know what's going on. Previously, we thought the messenger RNA, we could not explain this phenotype until when we look at the chromatin state, we're like, okay, when we overexpress FTO, we really opened up the chromatin state. The entire transcriptome is uplifted, all right? So it's really a way somehow open the chromatin state, and that somehow leads to a lot more cell proliferation at the root system. This is something I don't understand, but that's what's going on. And that drives the root development, right? Also drives more tillers, so you have more branches, right? So if you have a longer roots, the plant biology will tell you that's exactly what they want, right? That was signal to grow. If you have more branches, or leads to more biomass. So I actually had the rice today. Maybe some of you guys had a potato. So here's your potato, right? Obviously, there's out of 8 or 9 plants, 5 to 6, we see the stromatic effect. There's a couple, we're still trying to understand what's going on. What I want to say is that we're not adding something. We're adding a demethylase to remove a endogenously existing modification, right? So this regulation is intrinsic to the plant. We just help remove it. And this type of a chromatin regulation, the phenotype we've not seen with other systems so far in plants, at least, right? With that, I just want to close by saying that so far we've been working on M6A, and the lab has been spreading to these other modifications, to the uridine, very interesting, M5C, 2'-O-methyl, etc. Messenger RNA, and some of these seems to also occur on chromatin RNA. So we're excited, and hopefully next time we'll have more stories. I called the names. I just want to thank my collaborators and conflict of interest, and be happy to take questions later on here. Thank you.
Well, thank you so much. I think it's another example. We can see how better understanding of all these complicated regulatory pathways that don't necessarily change genes, but change their expression, the patterns of expression, can potentially have such dramatic effect on the organism. And I think hopefully soon we'll all be grateful for the new potatoes. All right, so at this point, I would like our speakers come here, and the speakers from morning session, Gary, if you would like, you can join as well. I think there were several unanswered questions also. Yeah, okay, yeah, let's also give an opportunity to ask more questions. I know there were more of them, and people got upset at me. I really wanted to ask the question, and so I had a warm-up question for Carolyn and Chuan. So if you were to look at the crystal ball, what you would, and you see yourself 5 years from now, what do you think the single thing you would love to understand about the genome? And
Well, of course, I'll have to personalize it to what I would like to see with our technology, which I would like to be able to get the better resolution and to be able to have the correlated fluorescence and X-ray, but the higher resolution of both fluorescence and X-ray. We have built a cryo-structural illumination microscope, so we can get a little bit better resolution than we've done with the cryo fluorescence tomography. Unfortunately, we have to write our own software because all of the existing SIM instruments use 2D software, and we need 3D software. We have to write some 3D software to do that. And then we have to figure out the way to be able to use the better resolution optics, maybe if it's just with a region of interest of the nucleus rather than whole nucleus. So that's one direction I'd like to go, is more correlative studies with actually multiple technologies, the fluorescence, X-ray, and then some cryo-ET as well. That's one thing I'd like to do.
Chuan?
Yeah, I'd love to see a spatial at the chromatin level, the correlation of imaging with sequencing. So like what showed this morning, this amazing change of chromatin state and what kind of a sequencing or chromatin features are there. I think at that scale, that would be amazing.
Right. Question over there.
Stuck myself in the corner. So in terms of engineering, I thought this last talk was particularly illuminating to one question, which is the difference between embryonic stem cells and iPS cells and the off state of chromatin, which I think is different between mouse and human. And it is, for instance, the inactivation of the X doesn't have the re-expression of the X doesn't happen in human iPS. So we really don't have the same model after re-derivation of a system to look at inactivation of the X. You can't do that in the human. You have to do that in mouse embryonic stem cells. So I'm possibly for everybody, but specifically for our last talk, might be an interesting question whether any of these tools could be engineered to make a better iPS cell in humans, so we could look at these critical issues of human development.
I assume that question is for me, right? Yes. Yes. That's a great question. We're approaching that way right now. We have some really interesting results. We can really editing the mouse embryo without touching the sequence of DNA, and we can make changes. We have a lot more success with plant right now, but the question is, how do you achieve the group effect? Because if you just added one gene, one chromatin state, one gene is not enough, right? And we dump FTO in, which affects the entire cell, right? Maybe some will be an advantage, some might be a deleterious. So we are using tricks to try to target a group of genes. For reprogramming, yeah, that would be very interesting. Look at the reprogramming. We're studying at a very low ground. We're looking at the stem cell right now. So our first task, we achieved the hematoponic stem cell. We can expand the ex vivo 10-14 folds. And then when we put this into transplant for 2 generations, we can still achieve 8 folds expansion of hematoponic stem cells. So it's a good step. Mesenchymal stem cells, the next step. I think the more we learn, the further we can eventually would love to be able to have a way to program the chromatin state.
I have one more question, Chuan, for you. So it's I think in your data you showed FTO cells after demethylation. You mentioned that they are resilient to drought, but you also said that they seem to be resilient to almost everything else, other stressors as well, right? What is going on there? All stressors are very different, right?
Yeah, they survive in alkaline conditions better. Like last week, my postdoc rushed to me, showed me the tobacco grow on sodium chloride. We survive better. I'm not sure that's a good news, but yeah. I think these plants, yeah, were growing cannabis, but we'll see. That's for PhD recruitment. These plants are just more robust. They're just more robust. They're, this can be a long session to discuss why, but we feel like there's this state of plants, right? Unlike human, we can run away from anything. Plants do not, right? Same plants survive in Amazon, in Chicago, Norway. There's the same plant. So we think there's this switch. We just turn them on if they're in Amazon, or lots of sun and when, which is somehow turn that switch on, we're still trying to figure out. And for that reason, they're just more robust.
Here. I actually have two questions for the first speaker. I'm a high energy physicist by training and biologist by trade, so two different questions. The first question is, what is the limit of your microscope? If it's redesigned, can you go to reach resolution of single nanometers in 3D?
The wavelength we use is 2.4 nanometers. So that's the theoretical limit. Yeah.
Okay, that was easy.
Yeah.
Now a hard one.
Reaching theory is not always easy.
A hard one. So you beautifully showed how, stem cells, the nucleus structure is so different from mature cells. And of course, then the question is, why don't we study that? And can you actually you mentioned word biopsy once, I believe, but you didn't show any biopsy. Can you make a biopsy from a heart or brain and actually freeze it in your pipette and do 3D reconstruction?
Theoretically, no, it's something we want to do. We have thought about that. We are making some, I don't think the glass capillaries have the strength, but we've been making some quartz capillaries. And I think the quartz have the strength to be able to do something like that. We're going to start with easier than biopsy specimens, but ideally that would be where we could go with that, is get some actual biopsy specimens. It's in the plan.
Yeah, I also have a question to Carolyn. Your X-ray really seems very remarkable. So it reminds me of a visualized version of NMR machine. So is it also possible to visualize and at the same time quantify the composition of the atoms so that we can actually do the NMR and the cryo-M kind of thing at the same time?
Yeah, I showed you the crystal of the oxalate. So that was one where we went forward. We knew the atomic composition. We calculated what the linear absorption coefficient should be. You can do the same in reverse, and that's what the modelers did with the pancreatic insulin granules. They worked both ways, knowing the composition of the molecule that they were looking at, determined the atomic composition and the lac value it should be, and took our lac values and worked backwards. So that was a really complex model that they did, going in both directions. And so it is doable. Not trivial, but doable.
So does it mean that we can also visualize the structure of the molecules, macromolecules as well, not just the cells?
Not just the what?
Macromolecules or polymers.
Visualize in what way? We're seeing the actual structure?
Yeah.
Again, we only, right now, actually, the best resolution anybody has achieved with soft X-ray tomography is 25 nanometers. That was just with a thin specimen. The best optics they've made are 10 nanometer. And they get tricky because what I didn't mention is that the nanofabricated device, the better the resolution, the harder they are to make. And not only do you need the rings being a finite distance apart, but it's the aspect ratio that gives you the sensitivity. And so the higher the aspect ratio, the greater the sensitivity, and also they get very floppy as you make them. So though we have this theoretical ability to go to 2.4, that's still not really good enough to see molecular structure. And I think that's going to be really challenging. We need some better optics.
So I'm a master's student. I'm sure there's some undergrads and some PhDs in the room as well. It's kind of a higher level question for all of you, but for researchers starting their careers, what advice can you give about how to make a career out of a passion, especially in a space like this?
That's a real tough one. And one thing I always say is, and you hit it right there with the passion, because without the passion, don't bother. I think you really need to love what you're doing. Passion is number one. How do you find that career that gives you that passion? I have probably one of the least linear pathways from where I started to where I ended up. So I don't think I can answer that. I think what I would say there is chance favors the prepared mind. look for the chances, look for the opportunities. And when you find something that hits you, somebody you're sitting in the audience of some talk, and that really strikes you as exciting, pursue it. Yeah. Anybody else?
Oh, I think you know, that I think encapsulates it, because basically it's, you know, I think any career in academics or industry or whatever is filled with challenges of different types. And it's just extremely difficult if you're not passionate about what you're doing. It doesn't mean you have to be motivated all the time. It doesn't mean that you know, you have to work 60-hour weeks, but it does mean that, you know, occasionally you're in the shower and you just start thinking about, you know, why neutrophils look the way they do, or, you know, whatever. And they send out these DNA curtains, so that's, but I think, yeah, I think it's, you know, I think there's a lot more. I think, you know, old days, we had the luxury of maybe not worrying so much about career paths. And I you know, mine was maybe a little more linear, but I definitely did not think in advance about where I was going to do my graduate work, where I was going to do my postdoc, what labs I'm going to, you know, I joined the postdoc lab I went to because I was visiting some other lab, and, you know, I went and said, oh, I just want to talk to this person. I just enjoyed talking to Alan Spratt. And I think, you know, there's a lot more emphasis today on planning out what your CV is going to look like and all this sort of stuff. But I think and, you know, I understand it's different, but I think at the core, it's still about, are you passionate about science, and do you think about it when, you know, you're doing other stuff?
I had never heard of a synchrotron when I was in graduate school. I had no idea what synchrotrons were about. So yeah. Yeah, no, just that. I think be curious, right? I mean, I think everything everybody's just seen here, and the thing about it as well, you heard it from Juan, you heard it from Carolyn, you heard it from Gary, even me, right? You know, people like to put barriers up, like, you know, it's this or it's that, but it's not. It's biology, right? You know, it's literally, it's physics, it's chemistry, it's RNA, it's DNA, right? And sometimes you can get really, really narrow, and then you don't see the rest around you, right? And so, and sometimes, like, again, you know, you could hear it in some of the talks. It's like, you know, these things keep bugging these people. It's like, why didn't it give me this really lethal phenotype, right? And sometimes you might need a technology or something and have to come back to that question. But it's the anomaly, right? You know, if the model doesn't explain everything, there's something we don't know. So don't force models onto things, right? Because, you know, and sometimes the answer is no, and that's an answer, right? Because it tells you what it's not, which allows you to discover what it is, right? As opposed to kind of following the kind of emperor's new clothes where everyone goes, is this, and actually when you look at these things, you go, hold on, that, you know, if that's the case, then this couldn't possibly be. These two possibilities can't coexist. So what is it? And that's how I think you kind of find, I think, things that are new. And I think the other thing that I would say is that fail fast, right? You know, it's if you can, like, if there's a Friday afternoon experiment, that's great. But kind of see if you can kind of fail quickly and do the last. I always tell my students, do the last experiment first, right? So just to speak to the competitive part, because I think, you know, one of the things that, you know, half the stuff, you know, that you might hear, you know, it's already kind of done. The next step is already done, right? But assume this, right? Assume that if there's something kind of obvious or the next molecule done the thing, right? What's the piece after that? And maybe go after that. Or if this is the principle, you know, and that's the discovery, well, what would you do with that information? And can you do that now? Then do it, right? Because then I think you actually get, it's more thoughtful. And if you don't do it, maybe no one else will, but it actually moves the field, I think, kind of forward, right? So to kind of don't be afraid to kind of go, well, if this is true, you know, you don't have to go from linear to here, but it's like, if this is this, and there's this general principle, then this would imply that. And that's, I think, how you really find central kind of
I couldn't help but notice there are several aspects that are fairly common across all of you. One of them is you are doing transformative research, disruptors, every one of you. But at the same time, your research also is highly interdisciplinary, or we can call it convergence. Even if you look at the career trajectory that you took, I think it's quite remarkable. Clodagh, you were trained as immunologist, right? Studying immune system, but your research clearly is much more broad than that. And we had a very vivid example of that in your talk today. Gary, you keep saying that you're a biologist, but at the same time, you introduced the concept of phase separation in genomics, something that until quite recently, only physicists could understand. Carolyn is a biologist by training, if I remember correctly, right? And you gave a talk that probably what nuclear physicists would be happy to give, right? And Chuan received his PhD in chemistry, right? If I remember correctly, from MIT. And the examples of his work include synthetic biology and molecular biology and genetics, et cetera, et cetera. So I think there are a lot of examples here of truly transdisciplinary research. So my question is, why is that it's so difficult to break the boundaries between fields, and what can we do to get better at it?
I think Gary hit the nail on the head this morning with communication. You have to be able to be willing to talk to people who speak a different language. I mean, I work with all sorts of people, mathematicians, physicists, modelers, et cetera. I'm curious. I like to talk to them. I like to hear how they do things. And it's just being willing to talk to people. The first time, Sam Isaacson, the mathematician who did the modeling of how molecules move through the nucleus, we were chatting on the phone. He was describing the data he was seeing. We had a Zoom, not phone. And I said, well, what does it look like? He drew out the mathematical equation. Waiting for an image, mathematical equation. So, it's just that first reaction was a little bit, this could be a problem. But, we've talked about it and we still collaborate. We have many projects. But yeah, what if you show me an equation?
I think the other component is to be fearless. And I think that we're often taught to stay in our silos. It's the safe bet. I know how to do this. I'll just keep doing it and doing it. And that's, to me, counter to essentially not only being passionate, but also sort of following what the science shows you. I think there's a difference between engineering and discovery-based science, right? And if you do discovery-based research, then you're going to be faced with things that aren't what you're expecting. And the question is, are you going to pursue that, or are you going to stay in your lane? And I gave one example, but this has happened to me over and over and over again. And I'd say what few breakthroughs we've had have all been these types of, wait, why is that happening? I'm always asking myself, what was I expecting, and is this fit? And especially with visual stuff, that's really a great way to go. So even the example I gave of that void, okay, we could just go, oh, that's really fun, and throw that out, but that is what got us to that protein. And we'll see what happens as time goes on, but if we had just ignored that, anyway. But I think you have to be fearless about going to places where you may fear to tread.
Yeah, I think I would just add, yes, I totally agree. So I actually think there's some great examples here based on my conversations over lunch, is that, so first of all, don't be afraid to say, I don't know, teach me, right? I don't know, tell me, right? You have to leave your ego. For me, cancer is a big driver, right? And I go, you know what? I don't care if you think I'm stupid, right? But I'd love to have an impact on a patient's life. If you can help me, I just don't care. Tell me, right? I really want to learn because I want to do that. And so, and I think that's a big fear, right? Where you go, oh, as a physicist, I don't know, I barely passed math, right? But they don't know as much biology, right? And so we all have, we all know different things, and surround yourself with, I think, people who are better than you if you can, actually. Again, this is an ego thing. You want people around you that are better than you, and have people in your lab that know things that are different to you, right? You can't possibly span everything, right? But I have people in my lab who know all these things, right? And that's amazing, right? Because then it's real synergy or a real team, right? Where they can do things I couldn't even possibly dream of. But we can start to, I think that's how you start to break things down, is having people bumping up against each other. One's a structural biologist, one's a computational biologist, one's a chemist, right? And we start to rethink, as you obviously have department structures, right? Classic department structures, which tend to have, if I have this many people all in the same place, that think the same thing. The problem is they may all think the same thing. And what you need is the disruptor who thinks about that differently, right? And so, because there's a tendency for people to kind of recruit kind of more of the same that kind of agree with them. And I would say, no, you want people who challenge you in a constructive way, and go, really, why do you think that? Where it's a genuine question, not a comment like, why is that? Because then those are the questions where I go, yeah, what? Actually, why do we think that? What is the data? And so I think, leave your ego at the door, that and always be prepared to learn and know that you know things and they know things, but together, then you get, I think, magic happening, right?
You said better what the point I was going to make. Be humble. Yeah, that's awesome.
Yeah, I will add, actually today is a lot easier, right? 20 years ago it was harder, but today I think to answer actually the first question, you need to have a passion, but don't just jump into the first thing you're passionate about. Stay open-minded. Or I tend to say these college students are great, but then the first scientists that they met, they decided to work on that. Maybe there's stuff beyond, right? And once you identify a good problem, you're problem oriented, then you seek out for help. You're humble. You don't know anything or everything. You just go ask for help.
Be passionate, be humble, be brave, right? Okay, that sounds like a really wonderful advice to train. Yeah. I can certainly learn myself. Oh, thank you. Well, I have a
question. I have a question for Clodagh, because I didn't get to ask. So in your star map, what are the chromatin filters at the peripheral that get pulled away? Do we know anything there special?
You mean at the peripheral?
Right.
Actually,
because those seems to be the key to get pulled, and then start to have this.
Yeah, well, so I think about the really come back. There's actually a lot of things,
right.
Respect to Condensates and lino. Hybrids and it's and again, right, essentially, I think it's like, how can you have heterochromatin against structural activation at the same time? Actually, these things coexist. But if we actually think about it, I need transcription to pass out of this. And so one of the questions is that, again, so if you actually sort of have organization, depending on how things go in town, you actually very well on this effect. In that, in part of good right, basically, or otherwise, I even don't think this would be done in this argument. That's not to kind of pull basically and kind of structure cells into DNA out, right? That has an effect then on surrounding genes, basically. Right. So, you need to think about that. But the other thing is that RNA starts to build up again at the periphery. You start to concentrate on this, especially these kind of things. They can start to drive almost their own sorts of homozygotic heterozygotic interactions, which then actually either in the non-linear way drive things forward. And also actually start to compress things on the inside. It's almost like you have different sorts of ionic or charge concentration. Right? So I think the you actually see some kind of particles kind of at the periphery, but there is actually just sorts of you might access that first, and it starts to kind of come unglued. But you start to build, it's not like everything opens up. So
Right.
then you start to sort of build up things, but these are motors.
Right. Wonder whether there are racial transposon elements there, introns there, or enhancers there.
No, yeah. So basically, as well as here, some of those particles that we see in the transgene, you can actually see on the periphery as well of kind of some chromosomes. So I'm not sure if that's to do with sort of an RNA decay method, basically, and an RNA decay method, or RNA. You get time to repeat or sorts of viral bidirectional. You start to get RNA DNA hybrids, even, right? We do get things that actually can actually sort of create actually then RNA boundaries at the periphery of DNA domains, which actually really give you different phases and actually different molecules. So I think it's a fascinating connection in terms of well of thinking about RNA, DNA, other basically neutralized, like not necessarily RNA. And how transcription in that itself can create the RNA that can feed back, because again, biology never wants to generate conditions apprentice. To turn on the issue, can't turn on, right? So I think it's really
I think we have to think about dynamics and about stochasticity. And so I think that it's not silent and active. It's not that there's a specific sequence out there that's doing this or doing that. It's that these are objects that are constantly in motion and not static. And so all you need to do is have one piece come out at one moment that the RNA polymerase can grab onto, and it. So I think, this is back to the original questions about dynamics. If you think too literally about it's either this or that, and not include the fact that there's a lot of dynamics and stochastic behaviors, and it's the ensemble that matters, right? Then at least for me, it becomes easier to think about how you can transition from one state to another.
Absolutely.
Yeah.
You have a virus coming in, or you have a signaling. Let's say you activate a, STAS3 or whatever. Then what's your landing path?
You increase the probability after.
Right.
I think one thing that's, again, really interesting, thinking about the unknown stress idea, right? Like PMO bodies, what are PMO bodies? So again, it's kind of the idea of valency, right? So a virus can be made far too quick, right? So, talking about question, in some ways it's sort of like, how do you counter that? But do you detect changes in what's called nuclear volume, right? Because as the genome kind of expanding into this, well, it's actually going to have really interesting effects, mostly biomechanically, on histones, basically, all these kind of things. So you're actually detecting changes in homeostasis, which is a universal signal of an RNA virus, DNA virus, right? And the other thing is some people, basically Carolyn will be able with herpes virus. And as far as the bicycle links, the mice come in, actually get silenced neurons. So it has this kind of up in this glitch and stuff like that, but it will actually, as opposed to 36 antivirus, it's right in the middle and get these PML bodies that see repetitive DNA, lying all over the NHC, all these different regions. The NHC looks right as well in these bodies, which are driven again by actually transposition. So like you set up these kind of sensory advantage from spaces that detect this kind of repetitive homotypic interaction together. Now, when they don't have necessarily any factors, but something when they don't switch to lots of elements, now you have a big that's really what you're being directed. Kind of like an IgM inside the cell.
Well, thank you so much, Chuan, Carolyn, Gary. Clodagh, thank you so much. It was a pleasure and privilege to have you. I learned a lot. I think everybody here learned a lot. And thank you. We still going to have a little reception with, I think there will be more food in case anybody's hungry. I'm not, but if anybody, we have a lot of food basically. You have it, you need to eat it. We can't waste food. No, it's the same place where the poster session is. I'll hope to see all of you there. Thank you.
Yes.
Stay curious and keep learning.
Yes.
Yes.
Thank you.
Yeah.
And on a